evening, everyone, and welcome back to the third and the last day of the 6th Advanced GMP Workshop 2021. Let's have a quick recap of what we had yesterday. We had great insights on recent advancements in freeze drying, continuous manufacturing, cold chain logistics, role of blockchain in next generation pharma, vaccine evaluation, and finally concluded with a panel discussion on digitization for quality and efficiency improvement in pharmaceutical operations. So let's get started for today. One of the key highlights of the IPA Quality Forum is the work of the IPA Quality Forum subgroups. Every year, 4 important topics central to achieving quality and manufacturing excellence are taken up. In this session, we will have the representatives of the 4 IPA Quality Forum subgroups 2021 share their knowledge they developed through this journey. Our first topic is sterile operations, covering aseptic practices, smoke studies, and media fill. We have with us Mr. Pradipta Swain, Vice President Operations Sun Pharma Limited, to have the session. Welcome, sir.
Thank you. Thank you. Good afternoon, good morning, and good day, everyone. Thank you so much, IPA, for providing me this opportunity to share our knowledge and work on aseptic practices and correlating that to smoke studies and media fill. Next, please. This is a general disclaimer. This presentation will reflect my own view and our view of the subgroup, but it doesn't represent any views or policy of the organization we are associated with or any entities. Next, please. We always wonder, anything scientifically we do, like aseptic practices, what is that, how is that, and why? So there will be some thoughts, there will be a source of contaminations, personal hygiene. Also, we will discuss thumb rule for aseptic practices, gown control, material control, and finally the correlations with smoke study and media fill. Next, please. So aseptic practices, all of us might have heard this terminology many times. What is aseptic practices? This is a set of behavior we all do to prevent contaminations into product, sterile component, or any critical parts by considering mainly microorganism, particulate matters, and any other contamination like pyrogen as well. To prevent any kind of contamination, whatever practices we do, that set of behavior is called as the aseptic practices. And how we do it? With the technological advancement, it evolves many ways to prevent contaminations. However, very important is our training, experience, educations, and most important among them is attitude. The attitude of the person governs how an aseptic practices will become excellence, and it will become a habit later. And why we do that? Ultimately, our medicines goes to patients. Patients has a high expectations from the medicines that they will get cure out of it. When we are talking about patients, that can be ourselves, myself, you all, and anyone in this world. So when we are working on patient safety, there is always the patient's life has to be considered when we are doing aseptic practices, and we should understand that any contamination can risk patient's life. Particulate matter can blockage their blood vessels, or any microorganism or endotoxin or pyrogen contaminations can risk the life of the patient. Next, please. This is very close to my heart. I wish to share that some thoughts. Let us not make it too difficult. On the left of the screen, you can see a person trying to walk on a rope with some pots on the head. Can we say it's an excellence. Yes, it's an excellence by practice, but let us not make it so difficult for the person to observe. So aseptic practices should be supported with design so that it doesn't become so difficult. In the same time, the next middle picture shows our risk-based approach. Even if we are going to a bathtub, the person is taking oxygen, having a good swimsuit, and taking a conservative approach. With use of the technology in aseptic processes to prevent contamination, we always must see and must have an approach of conservative, and with technological advancement we should always look forward to newer and safer technology. Finally, the attitude of a pilot very close to my heart. Whenever a pilot is flying an aeroplane, he keeps his or her worries outside. Passenger safety is his foremost importance, and a simple snag brings back the aeroplane to the ground, and in the same time he doesn't think it is a regulation requirement, a compliance requirement, or a audit requirement. He thinks passenger safety, safe take-up, safe landing, and passenger's life is foremost importance. In our case it's patient safety. So we should give importance to patient safety. If we follow patient safety, automatically the compliance will happen. Automatically, we will be always ready with safe procedure and we will not impact the product. Next, please. The source of contaminations, we all know. People are considered as the greatest source of contaminations in manufacturing of sterile products. The picture shows it all, and we should always try our best to separating operator from critical areas. And with the technological advancement, barrier technologies such as isolator, restricted access barrier systems are very helpful, and we should continue to use isolator and wraps instead of conventional process are no more useful, because we all know with availability of technology where we can reduce the risk, we must use those. Next, please. We all say aseptic practice should be followed at company or manufacturing suites, but I will tell it that it should start from our personal hygiene and our own home. We should always follow the hygienic practices at home as well, and the practices will followed at company will start from our home with personal hygiene. Next, please. When we call about personal hygiene, the important part is regular bathing and showering. Every day, preferably, I will tell twice we should do that, which will remove our skin cells as well as it will reduce the microorganism load on our body. And hand wash is a must. It will never replace anything, and sanitization is not equal to hand wash. So always hand wash must be done with the use of soap solution or any good soap after using toilets, after smoking, or after meal break, or during each entry to the aseptic area or critical area. So hand wash followed by sanitization, but hand wash is a must. Next, please. These are the thumb rules of aseptic practices. We all know no jewelry, no wristband, hand watch, cosmetic. During this festival period, we are very tempted to have these bands of God. Those should be, we should have a trust in God, but we should remove the band to save life. We should not have any cosmetic, keys, card, or personal medicine, and these are not allowed in aseptic area. No street clothes are allowed to the change room. Forget about the critical area. No street clothes are allowed to the change room leading to grade C or B area. Those street clothes should be removed in the primary change room, where we initially enter into factory premises, and then in the primary change room we should wear a clean garment covering the full body. One important thing, gloved hands are not sterile hands. With gloved hand, we cannot touch anything sterile. Always, to touch anything sterile or intervene, we must use a sterile contact part or instrument. So it should be remembered, gloved hands are not sterile. It is sanitized. Next, please. Aseptic interventions starts with fast air. Fast air means when the air is leaving from the HEPA surface or the ellipse, it should not hit any sanitized surface, and it should not hit operator body, and then it pass on to the sterile surface or critical surface. It should be always checked during the smoke study, and we must ensure that no air is hitting any sanitized surface or operator's body when we are doing any intervention or when we are doing aseptic operations. Contact of sterile material should be done with always with sterile instrument. We already discussed that. Next, please. In aseptic area, we need to move slowly and deliberately slow. Why? Because the unidirectional airflow, which is coming from the laminar airflow surface or the HEPA surface, if we walk faster, we disturb that unidirectional flow, and the floor level contaminations can come up to the working level. And in the same time, we also can shed particle into the cleaner air. So the slow movement should be deliberate by understanding how it will help us to prevent particulate and microbial contamination into the product, and no contamination should rise up to the working level. And this slow should be a practice. It should not be that so much slow that we cannot work. It should not be faster that it disturbs the air. The slow movement should be a practice. It should be same during media fill. It should be same during smoke study. It should be same during routine practice. So we should not defer these slow movement time to time, and we should not make it very slow when somebody is watching it. It should be a practice for aseptic behavior. And often I come across one word, robotic. So actually aseptic practices should not be robotic. Actually robot sex is body parts, hands, head. So but in aseptic practices we cannot shake our body part or hands. We should always try to minimize our movement of the body part and move slowly. Next, please. The operator during the initial induction and periodically must be shown the video of the airflow or smoke studies, and all intervention. We should not leave any single interventions to present it to the operator who will do aseptic actions, and must be trained to follow these actions during routine practices. The same aseptic technique and interventions must be followed exactly during smoke studies, successful media fill, and routine practices. And the position marking for the man and material on the floor, interventions, access point marking on machine, cubicle door, glove ports. For each type of interventions must be there. Example will come in next slide. Next, please. So if we see why to follow below aseptic practices with the marking of all these doors, position of the operator, position of the material, because through the smoke study, we always check that the fast air is not hitting any sanitized surface, and how the intervention is not impacting into the contaminations or the product or the critical zone. So if we do smoke through a glow pot for a particular intervention, we need to correlate that to media fill and also during routine operation. So there should be a list of the intervention with the door identification, glow pot identification, that which intervention through which door for which frequency for which durations, that should be marked, because operator cannot remember. And remember this, that we may mention that in batch manufacturing record, but operator cannot visualize the batch manufacturing record always. So it should be on the door, that which door allows which intervention and where the operator should stand during each intervention, and where the material are to be placed. And why we keep the forceps tip upward towards HEPA or LF, the first picture shows that, because we all discussed that the fast air should not hit the sanitized surface, operator body, or anything when it touches the sterile surface. The forceps tip is going near to the sterile item or sterile object. So when we keep the tip up, the fast air will hit the tip and it will not be contaminated. So similarly, door and glove port locations must be identified, as I mentioned, why it is, and marking position of the operator, and how the slow and deliberate movement with hands above working height. Why we should keep our hands above the working height? Because when hands are sanitized, if we put the hands down, it will go below the working height. If it goes below the working height, our all validations or area qualification or the contamination level are validated up to the working height. At floor level is a worst case, or the more contamination will be below the working height. So if we put after sanitization our hands below waist level or below the working height, then hands get contaminated. Hence, after sanitization in critical zone, we should keep our hands up as shown in the right hand side last picture. Next, please. So there are many speculations when we are doing aseptic technique or behavior, whether it is applicable or in isolator, whether we can little bit of relax for any aseptic technique. No. Even if with isolator, close restricted access barrier system, the aseptic techniques are always same, and aseptic technique cannot be relaxed in isolator. Aseptic interventions should always be from the side. So we should not do it from the top, because again the fast air will hit and go to the sterile surface. So we should do in a manner from the side, so that the sterile surfaces gets the fast air. And operators should refrain from speaking, because when we speak, we contaminate more with our speech. So we should refrain from speaking when in direct proximity to the critical zone or area. Next, please. The grade A machines, we should never use PVC curtains, because if we do PVC curtains, then that will create a challenge. We always should use hard partition, and when we use hard partition, it may open outside the grade A zone when we are opening a door for any intervention. Generally it doesn't happen, but if it happens, then we must do sanitization of the inner side of the door, and before we are closing the door. So we also should understand there must be a grade A continuity from the point of sterilization of any item. When it goes to a zone for assembly or usage, it should always travel through grade A continuity, and a mobile cart can be used for that, and we can also use a grade A zone for that. For isolators, all the components are to be sterilized in autoclave or with the CIP SIP system of the isolator itself, and it should be transferred to the isolator. Here the point is, when we are doing aseptic intervention in isolator, do not perform it before decontamination cycle. We have to put all the items without opening the contact surfaces to the extent feasible for a better placement with a closed door, but we should not expose the contact surface during the disinfection cycle of isolator. Post to decontamination cycle, we can remove those wrappers or the tip wrapped with bacterial barrier paper through the glove pots. And we must ensure these wrappers should not go and block the risers inside the isolator, and it should be placed in a bin which was provided inside the isolator. That also we need to take care during the aseptic practices inside isolator. Next, please. So when we talk about people are the source of contaminations, so people entry to critical zone must be restricted to qualified and authorized people only, and it must be minimized. Each room should be qualified with number of people presence, and we must qualify that with the equal or worst case. Suppose an aseptic area allows 4 people, we may qualify that with 5 people, but not 3 people. So always the dynamic operations or the NBPC count must be done with the personnel present with activity on, and number of personnel is important during qualification, including media field. So when we are restricting the people, we must monitor it as well, that who is going where, and record it and review regularly. And we can do it through the biometric record or electronic smart access system available now in worldwide. Next, please. The frequency durations of each aseptic interventions must be restricted, monitored, and recorded and reviewed. Here I told 3 things: type, frequency, and durations of aseptic interventions. All interventions must be simulated or conducted in airflow pattern, and as well as in successful media fill, and based on which it should be controlled through procedure for its type, frequency, and durations. Electronic intervention recording systems are very helpful in this, because operator may miss and operator may not get an alert that how many times he did, what duration he or she did. So with the electronic intervention system, with alerting the operator for the time and type and which interventions will be helpful. Next, please. So we already discussed frequent sanitization of hands above working height, and why we have to do. We should not touch any wall or surfaces by our gowns. Next, please. So when we are talking about the gowns, the important part is operator must know the external part of the gown should never be touched, and operator should not get engaged in any activity which can contaminate his or her gown, like cleaning activity. Once the operator is doing cleaning activity, he or she should come out, change the gown, and then go in. Any maintenance, we should not continue working there. We should come out, change the gown, and work. And only persons who are appropriately qualified for gowning will be permitted to the aseptic area, and the person who need to do interventions in grade A must be qualified in the successful media field. For gown use, we must follow a standard mentioned in IEST, Institute of Environmental Science and Technology, that is IEST RPCC 003.4. And last but not least, about gown control, the goggle, many times we use perforated goggles, that is directly opening to the environment. That is not acceptable. All the goggle must be indirect ventilation type as shown in the picture. Next, please. For material control, anything entering grade A zone must be sterile or adequately sanitized if sterilization is not permissible. And then the material should pass through different cascade, like D2C, C2B, and finally B2A. If cascade is not possible, then a VHP pass box can be used, and E-beam tunnel rapid transfer port can be used for the same. Next, please. The correlations of aseptic practice with airflow pattern and smoke study, we already discussed it before. The risk assessment of aseptic process must be done. We must know what is the type, frequency, and duration happened in last 25 batches or in last 6 months, whichever is higher under a protocol. And then these aseptic practices for a new facility can be done through brainstorming and similar line or similar practices what is there. Then the water fill can be done. And when we mention the type, durations, and frequency through the water trial or experience of the line or a data collected for the routine media fill, then we should go for a smoke study for a new line, and based on the this type, duration and location, that has to be simulated in smoke study. And post successful smoke study, we should do media fill with the same type, durations. And the difference is, in media fill we have to also simulate the frequency, number of time, but in smoke number of time is not required, but duration of opening is required. Next please. So one important note, when we are performing any aseptic interventions, we should not do excessive interventions by telling we qualified in media fill. High risk operations should not be justified with successful smoke study or media fill. And use of the best technology, design and facility reduce risk only if with aseptic, appropriate aseptic practices are performed. Excellence should be a habit. Patient safety is our ultimate goal. We should never say compliance is a requirement or we should be audit ready. We should always try our best to reduce risk to the patient life, even if there is no audit, even if there is no observation, even if there is no supervision. The patient safety is foremost. If we follow patient safety and risk based approach, compliance will happen automatically. And in the same time, we should make our plant always patient safety ready, not audit ready. Next please. Thank you so much.
Thank you, sir. I invite Shiri sir for the question and answer session.
Thank you, Pradipta. It was very crisp presentation, covering almost all the aspects. And it is, I know it is impossible to cover every aspects of the aseptic in just half an hour. I understand that. I think there is one question which has come. So during media field, each media field should have all the interventions, or three media field all together should consider all the interventions which are happening? What is
good question? Thank you so much. So during media fill, we need to have 100% of each intervention, routine and non-routine, in each media fill. We cannot segregate those intervention into different runs of the media fill. It has to be performed in all, because we want to see the consistency of those interventions in multiple media fill. So all intervention which are routine and non-routine should be 100% performed in each media fill. However, worst cases like hold time, increasing the number of people to the maximum during filling, these can be staggered between the media fill. All hold times should not be simulated in one media fill, because if there is a challenge for any worst cases or hold time, we will not understand due to which hold time or which worst case there is a challenge in the media fill. So worst cases can be rotated. However, routine and non-routine should be 100% in each media fill.
There is one more question. Like in the solid orals, the batches are continued without really cleaning the equipment. 5 batches are kept. Can we do the similar thing in the sterile operations? Without autoclaving, can you go to the next batch?
I already told during my presentation, high risk operations should not be qualified in aseptic operations. So if based on our risk evaluations, there is a risk to the patient safety, then we cannot proceed with that. We cannot go for campaign instead of sterilization and full cleaning. In aseptic practices, we have to be conservative and we cannot have those kind of campaign approach.
Thank you, Pradeep. I think that's all questions from the audience. We are just waiting for your guideline to come for the publication. That's what last thing is remaining. I think within short time it will happen. Thank you.
Thank you. Thank you so much.
Thank you, sir. The aseptic practices have been explained very well in a simple and a lucid language and with live examples. I'm sure this is an easy and a good learning for all the participants. Before we move on to our next set of speakers, I urge all the participants to share in their questions in the chat box. It's very, very important. It will be a virtual live platform. That's very important for us to keep this session going. Our next set of speakers will cover on cleaning methodology and validation. Please welcome Mr. Mahendra Singh Charan, General Manager, Corporate Quality Assurance, Zydus Cadila, and Mr. Santosh Kumar Yadav, Deputy General Manager, Quality Assurance, Zydus Cadila. Welcome, sirs.
Thank you. Namaskar, good afternoon, and greetings to all. Myself, Santosh Kumar Yadav, and I am joined with my colleague Mahendra Singh. Thank you very much, Indian Pharmaceutical Alliance, for arranging this Advanced GMP Workshop and giving us an opportunity to speak on this topic, cleaning methodology and validation. Though we are representing this best practice document, however, it is an outcome of great efforts taken by key pharmaceutical company associates who were involved in brainstorming, collaborating, and writing this best practice document. The team involved Mr. Navnath Pawar from Sun Pharma, Mr. Krishna Mohan from DRL, Ms. Gargi Sharma from Lupin, Ms. Tripti Gandhi from Torrent, Mr. Sanjay Gare and Mr. Nilesh Ebu from Cipla, and Mr. S.B. Gopalakrishnan from Zydus Cadila. And not the least, we would like to mention special mention of Mr. Silish Verapure, who from IPA, who facilitated this initiative and took it to the final stage where we are. Now, with due respect to all the seniors and delegates attending this session, we would like to start the session. Cleaning validation has been the most evolving and debated topic in the past couple of years, and this topic has been in limelight in different pharmaceutical industry with respect to the FDA citations. A lot of work has been done altogether, and several best practices followed across the industry has been collated and put into this best practices document. We feel privileged and proud to know that the best practice document is now ready, and the book copy of which was shown by the Drug Controller General of India, Mr. V.G. Somani, on the very first day of this workshop. Next slide, please. Yeah, thank you. As we are aware, there are several guidances available on the cleaning validation from various regulatory agencies, and all the leading pharmaceutical companies are meeting the expectations. This IPA best practice document provides an hands-on experience, hands-on approach to support the pharmaceutical industry, and it is not intended to interpret or replace the GMP guidances provided by any regulatory agency. Next slide, please. Yeah, as we are aware, there are several guidances available on cleaning validation. The team worked on this best practices document while reviewing the having a gathering meeting on this topic. It was noted that there are different approaches adopted by different companies, which we shall discuss in the subsequent slides. And it was noted that more than one approach was seen across the industry, but all were found logical and acceptable. Below are some potential areas of improvement which were identified during these meetings, which included multiple approaches for worst case selections, the traditional approach towards the life cycle approach, and logical grouping of product and equipment matrix for identifying worst case, risk-based approach for developing cleaning procedures, and also identifying the hard to clean locations, bracketing of equipment with respect to the size group for developing cleaning procedures, handling of potent molecules in the shared facility, handling of failures, qualifications of operators, inspectors and samplers, management of data using software-based application, and statistical evaluation of cleaning verification and validation. Some of the key aspects we shall discuss in the subsequent slides. Next slide, please. Now, this slide presents the comparison of different companies across the columns with respect to different approaches followed. The approach included worst case selection, the MACO, with respect to API and the cleaning agent, the grouping approach. For instance, we can see that in worst case selection, some companies are following the solubility approach, that is least solubility. Some companies are following the cleanability approach, and we can also see that there are other companies which are following the risk-based approach, wherein risk priority number is calculated based on different factors of API solubility, potency, and toxicity. Similarly, for MACO in API, we can see that the companies are following the PD approach, and some companies are following most stringent of dose criteria, 10 ppm criteria, PD criteria. And with respect to MACO cleaning, again the comparison is showing that there are variations, and it is with respect to PD and LD50s. And for the grouping approach, again for product and equipment grouping, it is varying from company to company. Next slide, please. So now this slide again looking to more parameters, the bracketing approach on the equipment size group, especially on the bracketing of equipment groups. When we want to make a group, what size we can take into consideration to group a equipment for which we can have a single cleaning procedure. So this was also taken into account. The statistical evaluation of cleaning, verification, and validation. Few companies had this, and others are still making. So coming to the last one was on management of data using software. So still few companies had the management of database through software. Slide please. This slide shows the shift of the traditional approach to the life cycle approach. As we know, the FDA guidance of 2011, similar approach for cleaning validation has been adopted, and it has in practice are using three phases. The phase 1 has the cleaning process design. The phase 2 has cleaning process qualification. The phase 3 is continued cleaning verification for ongoing monitoring. And very important note here is during each of these phases, the knowledge gained at each phase of the cleaning validation, it can serve as the feedback or feed forward mechanism for the life cycle approach in the management of cleaning process. Slide please. Yeah, now this slide we are talking about the multiple approaches of worst case selection. So one of the approach, acceptable approach, is worst case based on the cleanability, wherein we can do a formulation assessment, the scientific formulation assessment, wherein we can consider the solubility criteria for API as well as excipient in the entire product composition, and the insoluble load on that equipment in terms of kilogram will be converted, and similar matrix for all the products. Once it is made, you can derive to a worst case product having the product which is having the highest total insoluble mass on that equipment becomes the worst case product. Next slide please. Now the second approach, which is before I go to next approach, in approach 1 under cleanability, there is also a coupon study available wherein you can perform in the laboratory using different coupons with a known product concentration of different product, and you can see how long particular after cleaning, how long the product takes, the cleaning of the product takes. Based on that, cleanability assessment can be done using the coupon study. The product which is taking the largest time becomes the worst on the coupons which are prepared for different products. Now talking about the approach 2, which is on the risk prioritization. So risk prioritization, we can see that it considers different factors which are later rated and to obtain a risk priority number. The factors included here are lowest therapeutic dose, that is SRDD, then the PD or AD, which is the lowest PD, will be taken into account, solubility of water or cleaning solvent, and the cleanability approach based on the operator production feedback. And you can see the first column below table has risk factor, which can be applied to obtain a risk priority number, and the product which is coming as the having highest risk priority number becomes the worst case as per this approach. Next slide please. So on several aspects, the clarity of concepts and technicalities has been included in the best practice document to facilitate the understanding to the end users. In doing so, one of the key important section is how to develop and write cleaning procedures using quality risk assessment tool. So this section will explain, we are going to explain one example. So in doing so, the quality risk assessment tool, the key components of a tool should include, risk assessment should include the tool selection, identification of the cross-functional team who will participate in execution of this risk assessment, equipment description, equipment operation and cleaning process, whether it is manual cleaning or automatic cleaning, then major components and subparts for contact and non-contact parts, schematic diagram of the equipment, identification of potential failure modes for contamination or cross-contamination, areas difficult to visually inspect without dismantling, identification of powder adherence probabilities on high risk indirect product contact surfaces, which includes duct, drains, lower plenum, etc. Next slide, please. So continuing, component-wise risk assessment should be done to identify potential failure modes. So we have to break, based on the dismantleability, we should break the different components of an equipment and do a potential failure mode for each of those components. Risk management preparedness should include a thorough Gemba walk to understand the assembling and disassembling or dismantling of the equipment in view of the cleaning and the probability of deposition of the product residues, and the interview with shop floor people to understand the existing cleaning process and to identify the process with respect to enhancement actions. Historical review, doing a historical review for OS and deviations, particularly for cleaning validation and line clearance failures. So this will help in understanding what are the areas of focus, and also for doing a probability of occurrence rating, we can review a retrospective review of QMS events can be done. Right, please. Now this slide shows a snapshot of FMEA template which can be applied for doing a risk assessment. The elements can include equipment components, subparts, the product non-contact, contact parts, failure modes, and then rating to arrive at RPN. And the last column has a risk mitigation plan against the action plan. So overall, after the risk assessment is complete, the expected outcome is the conclusion and final recommendation, which can include precise instructions for dismantling and cleaning of subparts for a given equipment, defining types of cleaning aids which will be required for cleaning of different equipment parts, scrubbers, brushes, scrapers, etc. Defining aids to examine visual cleanliness checks for components and parts equipment using illuminated torchlight or GoPro camera in the indirect contacts like confined areas like ducts. Exploring engineering solutions for cleaning of large surfaces or ducts. So this has been in the recent past, we have several observations of ducts. So going forward, looking for an engineering solution can be a WIP solution, WIP system, which is wash in place system, or a CIP system, which can be used as a WIP skid connected from water source, and then we can have the sequence of cleaning operations. Let's say, for example, of FBD, we can have it for exhaust duct cleaning sequence, then the inlet duct, and then the body part of the FBD. So that can be a recipe-based, engineering solution which can clean the duct which is not reachable by manual mode. Next slide, please. Yeah, so this slide we will discuss and share one example of an FBD checklist. Before that, I will just summarize the key features of this checklist. Is like it will have a proper chronology of sequence of cleaning steps, which includes initial dry cleaning and dismantling, then pre-wash or initial wash with the process water, and then scrubbing of different equipment parts which needs to be scrubbed for proper cleaning, then washing of equipment parts and indirect contacts, including ducts, initial inspection to observe that cleaning has happened, and then finally putting a purified water rinse, followed by drying, and final inspection of the equipment, which will be done in the dismantled form, so that you are able to see all the parts of the equipment, whether it is clean and certified. So the checklist which is designed has a human error reduction format, which we will see and understand, which has important element of important instructions being highlighted in bold, italics, and critical process parameters are annotated with special symbol, which is like attention activators or attention indicators. Then pictorial diagram of individual parts to understand and correlate what is that in the checklist. And with that, we have a display of key components in the checklist and cleaning aids. So can I request to open the checklist? This is the embedded file. If you can open it for, and then I will be requesting for scrolling it down just to have a view of this checklist. Yeah, can we open the checklist which is embedded as file? Yeah. Yeah. So we'll move ahead and probably we'll try to see this towards the end of this session. So yeah. So this slide talks about grouping and bracketing approach. The industry-wide approach followed at the leading companies has been brought into one place. The product grouping has important two sections wherein we can do logical grouping of products using different facilities, product mix, or dosage forms. And like dosage forms, you can say the oral solids, you have general category products can be made as one group. The potent category can be another group. Then other dosages like aerosols, nasals can be a different group. Okay, like animal products can be of different group. So that way dosage form can be made, or if you want to group as product facility, if you have multiple facilities, you can have block wise or facility wise product grouping done. Second option in the guidance given is of surrogate product. So surrogate worst case product, I would like some time to focus on this. This has been mentioned in PDA TR 29 as well, the guidance. Surrogate worst case product is a specially formulated product which may not be a commercial product, but it becomes a, we are creating this product, so it becomes a worst case product in all the aspects which are than the routine manufactured products. From all the routine manufactured products, this becomes the worst case. And the features, salient features can be in this. The ingredients which are used should be easily available and at low cost. The properties of this, since it is becoming a worst case, so properties of solubility properties of this product, the ingredient should be insoluble in nature. Product composition should include polymers which are difficult to remove and also susceptible to microbial growth. And then formulation can be wet granulation, having a low limit of detection using PLC method or something which can be a faster detection and a low disposal cost. The rationale behind this approach is we can maintain the continuity of worst case product, because it has been seen that in many times we find it difficult that what we have identified as worst case product is a low volume product and it is not frequently manufactured due to its low volume requirement or poor market visibility. So in all those cases, this surrogate product can serve as a purpose which is more worse than any other product. And then another rationale to have this is to minimize a situation in which new worst cases products are added. So it can also take care of new product which is getting introduced, and if it is not worse than the surrogate product, then I have the validation still valid, and you can only do the verifications for introduction of this and then can continue. The only disadvantage can be the product manufacturing cost. You have to develop the product and method with respect to its manufacturing and the disposal cost. So next is the, please be on the previous slide. Yeah, second is on the equipment grouping. So on the equipment grouping, the guidance has provided clarity on grouping of equipment with respect to its complexity and size of the equipment. The equipment size, we discussed that 5:1, not more than 5:1 can be grouped as a logical grouping for developing a cleaning procedures. Then also we, if whatever is not falling under the worst case, assessment, those equipment or equipment group needs a confirmatory run. That is, we need to perform cleaning verification run on those equipment to say that it is evaluated or assessed. In large equipment surface areas, the worst case validation should be performed. And as a common approach, industry-wide, it is combining product and group equipment grouping is done, which is followed, and rationalized worst case products are taken for evaluation. Next slide, please. This slide explains about the potent molecules handling in shared facility. So we know that potent molecules ideally should be prepared or manufactured in a dedicated facility, but wherein it is shared facilities are used, we need to take care, special consideration should be given with respect to the personal protective equipment for operators, and also a special cleaning, additional cleaning of the equipment. There are generally two approaches followed in the industry, wherein after manufacturing of potent molecule or potent product, a placebo batch is run between the two cleaning processes, and subsequent it is available for the subsequent manufacturing. The second approach is to do a 2 times cleaning, wherein you will do exactly the same cleaning second time. While doing the validation, we need to establish that after first cleaning also it is good enough to go, while the protocol or acceptance criteria will be always after the second cleaning. During the validation, we need to establish by taking swabs after the first cleaning and also after the second cleaning, and the results should be compared. Both the results should meet the macro requirement. In special cases where potent molecules are manufactured, it needs or it is recommended, strongly recommended, that we need to do a routine monitoring of these products. So once the validation is established, what we can do is we can do a routine monitoring on reduced sampling. That is, if I say, for example, if I take the sample size on an equipment is say number 10 swab locations, in routine monitoring it can be reduced to 1 or 2 based on the worst of worst location results. The data which has been generated for validation should be considered for review. Whatever worst location data is available, those should be taken for routine monitoring. Now, this is important with respect to the qualification of cleaning operators, inspectors, and samplers. The guidance includes the requirement prerequisite on how to qualify an operator, cleaning operator, who has some prerequisites to complete, like eyesight checkup, the basic trainings of the cleaning procedures, and then the certification, independent certification of 3 cleaning cycle runs by supervisor and the quality assurance certifying it. For visual inspectors qualifications, there are factors and tools given, which is important with respect to qualify, of course after training, but they have to be qualified how they are doing the surface inspection. The factors or tools should consider distance of viewing, angle of viewing, lighting type, then the tools which are used for visual inspections, torch, GoPro camera, etc. And also it needs to evaluate the critical surfaces, that is hard to clean surfaces, while doing the inspection. The sampler qualification, which is a swab sampler qualification, the person should be again qualified using the coupon study in laboratory for a known concentration 3 times, so that the person is meeting the criteria that has been guided in the best practice document. Right. Now this slide talks about managing the cleaning validation database using software applications. So the software which will be used, which should be qualified and validated, and generally it is very helpful for a large manufacturing sites which has large number of products and equipments. So some key benefits of software is the data management becomes very easy with respect to product and data, equipment data. It goes for paperless protocol and report preparation. Online study execution is possible, like online punching of sampling and reporting. All the change assessment, like addition, deletion of equipment, new product, or impact on existing worst case, impact on existing macro, all gets evaluated through the software automatically. And ease of data retrieval, online tracking of the periodic cleaning verifications, the data trending is possible, and at the end it is 21 CFR compliant, the data reliability. Next slide, please. Yeah, on statistical evaluation of cleaning verification and validation, this is important with respect to the phase 3, what we discussed. Phase 3 of the cleaning validation life cycle, that is ongoing verification, continued cleaning verification. So these alert and action levels may be derived based on the data available for number of products on a cleaning procedure or for cleaning equipment. So it helps in understanding the general trend what we are against the macro value, so that we always have the scope to review the drifts observed in the residue results, and it can give a capability evaluation, and it helps to understand whether the effectiveness of manual cleaning, cleaning procedure, test methods, cleaning aids, and upkeep of equipment, especially wear and tear of the equipment, also gets evaluated at this point. It can always help and trigger an investigation or revalidation case specific. So that helps in if it is possible, you can show the cleaning checklist which I was asking for the embedded file. Okay. So with this, I will conclude my presentation. Thank you for patience listening.
I invite Sirisa for the Q&A session.
Thank you, Santosh, and for a very nice presentation. I was the part of this group, so I am aware what has gone into the details which we have gone into. And there are some questions. First question is from Jagdish Jain of Sun Pharma. He's asking, what is the meaning of surrogate worst case? Can you explain?
So if I have heard it correct, what is the minimum criteria for a surrogate product?
The meaning of surrogate worst case. You mentioned presentation, right?
Sorry, sir.
Are you able to hear me?
Yeah, little.
Sir, can you be a bit louder?
Surrogate worst case. What is the meaning of surrogate worst case?
Yeah, sir, I will add to it. Yeah. So surrogate worst case product is an artificially constructed product. It is not the routine manufactured product. But what we are saying is surrogate worst case product can be developed by a site, by an organization, which can serve as worst of worst product across the worst of worst from the products group which is manufactured at a facility, and then that gets evaluated with respect to the cleaning validation. It is a worst of worst product created which is not routinely manufactured. So in those cases, we need to manufacture that product. Then method needs to be developed as per that requirement, and then we do a validation. So it will serve as single product across the facility for worst case evaluation.
Thank you. Second question is from Kishore Shah of Sun Pharma. Ongoing cleaning validation, verification. How one can ensure cleaning by just taking a small amount of sample?
Yeah,
Are you
so
able to
on
hear me?
verification, there are multiple factors here. I explained about one of the periodic verification, but yes, first thing is the visual cleanliness is mandatory, and in every batch we do that visual certification of equipment cleanliness. Second is we should do periodic cleaning verifications, which includes maybe annual assessment of cleaning the equipment for worst cases. And third is like periodic monitoring, wherein we are saying that we have to review the changes, the deviations, the equipment wear and tear with respect to the with respect to the cleaning of equipment, and data trending, which is part of whatever data has been generated for an equipment with respect to the swab results. All those data for a particular equipment which is used using a single cleaning, cleaned using a single cleaning procedure, those can be populated to have trending evaluation done. So all these factors can be considered for routine periodic monitoring.
Thank you, Santosh. Last question. Now the newer and impurities are coming. So do you expect that like nitrosamines and other things, the estimation of those impurities during the changeovers will be requirement over the period?
Yeah, it's a very good question, but yes, impurity, as we know that nitrosamine or genotoxic impurities, these are things which are still in the pipeline, but initially we need to do a complete risk assessment wherein we need to rule out the process and equipment which we are manufacturing the products. Those products should be free, or else if we are having those indicators that there can be a nitrosamine generating product, ideally those products should be made into a dedicated facility so that we don't have any question on impurities.
However, as of now, nobody is actually, I think.
Yeah, in formulations, as to be precise, as such, we are not having any, so far action plan to have nitrosamine impurity being checked, because we need to have that method and further assessment. As such, the product is evaluated at different stages, including stability. But yeah, with respect to cleaning validation, it is
As of now.
yeah, correct. It is yet to be established. Recommendation is it can go for a dedicated facility if it is really area of concern.
Thank you, Santosh and Mahendra. Thank you very much.
Thank you,
No
sir.
more.
Yes.
Thank you, sirs, for the insightful session on cleaning methodology and validation. Our next speaker is Mr. Sanjeev Sharma, Engineering Projects and Maintenance from Syngene International Limited, who will cover the engineering and facility management. Welcome, sir.
Yeah, hi everyone, and first of all, let me thank IPA team and Mr. Bellapure for giving me this opportunity to talk in this forum about the best maintenance practices. As you know, the maintenance as such plays very important role in any upkeep and business of any organization. Now, primarily, there are different maintenance practices which are prevailing. One is, of course, the baseline practice where it is mainly done for meeting the requirement and of the CGMP based on the practices, based on the set procedures where you ensure that the drug is intact and it is not affected due to lack of any maintenance procedures in place. Then there's a maintenance good practices which does not have direct impact on the system, but of course it obviously is encompassed in the maintenance baseline practices, and the proper documentation practices has to be maintained. Now, when we come to maintenance best practices, it is beyond the maintenance baseline practice, which includes the different tools which will enhance the practices, and like we'll have a proper reliability centered maintenance, we'll have enhanced controls, trending analysis to be done, benchmarking of the system is being done. And once everything is finalized, then the maintenance program is rolled out, which is with approval of QA assurance, quality assurance, and then it is rolled out with with the proper systemic procedures and to maintain the quality of the product. Next, please. So these are typically the maintenance philosophies which we have prevailing. So one of these, of course, the breakdown and emergency maintenance, which means the breakdown maintenance or emergency maintenance is more or less called as a run to failure maintenance, where we wait for the breakdown to occur and then the maintenance is done. So this has, although the cost is practically nil in this, I mean, it's only when the breakdown of the equipment occurs, the people are attending that. But until there's a major breakdown, you'll have a major effect on the productivity of the equipment. Then the second thing is the corrective maintenance, which is kind of just in time maintenance, where again the maintenance is being done based on the symptoms which are the equipment is giving. If there's like if there is a abnormal noise coming from the equipment, you start attending the equipment before it is before it breaks down, and then attend the equipment and bring it to the normal running system. But then it's still not recommended because it's very close to having a breakdown in place. Then the third thing, which is most of the companies, I mean, is prevailing and is a set procedure, even approved by the QA, and it's a standard procedure which we do is a preventive maintenance, where the preventive maintenance of any equipment is done in the predetermined intervals and with the prescribed criteria. The checklist is derived just to reduce the failure and make sure the equipment is up and running most of the time. Next slide, please. Then the other approach is risk-based maintenance, where the equipment trending is done, the analysis of all the equipment parameters are completed, the periodic test of the equipment is done, and basis which the asset condition and the risk assessment is defined with appropriate maintenance program is also derived from there. So all equipments, displaying the values are kind of checked and assessed to give the like useful life and guarantee of the equipment, which has a high reliability and the safety and efficiency of the plant. The other most important aspect is condition-based maintenance. Now this is the most sought for, and it is coming into the practice now, where the equipment is the performance of the equipment is measured and the control of the corrective actions are being taken. So there is an online detection of real-time parameters of the equipment like vibration, sound, and then based on that, the equipment maintenance program is arrived at. So this gives very early indicators, or if there is a vibration, definitely it gives early indicator, and basis which the equipment is attended, and leads to a longer life of the equipment, and thus the productivity is also enhanced. Next slide, please. Now, as we go, and obviously one, we cannot manage if you're not measuring anything. So what we want to achieve from these maintenance practices is definitely to reduce the breakdown, improve the mean time between failures, reduce mean time to repair for any equipment, then reduce the maintenance cost. Obviously, once the equipment is maintained properly, the cost of the maintenance also comes down. And then, of course, with that, the increase in utilization of the equipment happens, and maintenance skill is developed. And of course, these all helps us in optimizing the spare parts in the inventory. Next, please. Now, these are some of the different tools which are kind of are in place, where most of the companies have started using this. One is, of course, RCM, that is reliability centered maintenance. Root cause analysis is being done. There's a Six Sigma practice. Then there's a Kaizen, which is happening. FME is being done on the equipment during installation, as well as during to arrive at the different failure modes of the equipment. Then the value stream mapping is done. Error proofing is completed, and 5S is, of course, one of the most prevalent practices which is now happening in industry. So these all practices, kind of increases the life of equipment, improves, enhances the productivity of the facility, and of course improves the uptime of the instrument equipment itself. Next, please. Now, when we talk about root cause and reliability centered maintenance, so root cause is something which has happened in past, and we investigate the root cause of the failure and arrive at the preventive action to avoid further happening of this the same incident. So it is more of a reactive maintenance, whereas the reliability centered maintenance is aimed towards going futuristic and understand what could happen, what could go wrong with the equipment. So it is more of a developing a maintenance strategy, and to ensure that the equipment and the process functions are in accordance with its design parameters, safety, and reliability capabilities of the equipment. So this is more of proactive measures which gives clear visibility to the maintenance engineers. Okay, this is what can go wrong with the equipment, and these are the important failure modes in the equipment, which we can address it on time. Next, please. Now, generally, root cause analysis is done on the equipment, but by and large, sometimes people they skip that. They just attend the symptoms, and the root cause is left as it is. So, like you say, in this, one has to really answer why we are replacing the bearing every time for the same equipment. Is it because there is vibration in the equipment, or is actually the root cause is a misalignment? Because most of the failure which we have seen in equipment bearings and all, that is because of the misalignment. So just changing the bearing on failure does not help. It's important to really get into the root cause, understand what is happening with the equipment, check the alignment of the equipment, although it is taking time, and then replace. Then only the further action should be taken. Otherwise, we'll end up changing the bearings again. And same is the case with the air compressor. So you frequently see the air compressor being loading and unloading is happening, and there's a continuous consumption of air in the system. Now, we generally see, okay, my air compressor is falling short of capacity, but is it is it the only thing, or do you have any leakages in their line? Because generally, even the leakages, small leakages, they come up to maybe more than 100 cfm or 200 cfm in the system of the large plants. So it's important to routinely check the leakages in the air, and make sure the leakages are arrested, so that you also save on the power, and you also save on the capacity of the equipment which you have in there in air compressor. So these are some of the places where the root cause analysis for any failure or for the cost saving, it plays an important role here. Next, please. So some of the tools and strategies which are being used, like if you see the screening tools, like there is these vibration analyzers are being used, then thermography images are being taken up to make sure that whether there is any heating up in the electrical panels and all that. And vibration sensors also used to see whether some vibrations are coming up in the equipment, which will kind of lead to vibrations. So these are the predictive tools which plays very important role, and continuous monitoring of vibrations, sound, thermography images of in electrical panels. Those have to be done to make sure, and even harmonics checks in the panels has to be done to check, make sure that your system is in robust condition, and you don't have a breakdown in the equipment as such. So these are some of the tools which any maintenance team should have to make sure that their equipment are continuously monitored and maintained. Next, please. Now, this is typically RCM flow, which is, like you first try and identify the critical equipment basis, whether the equipment has a lower MTBF, whether it is bottleneck equipment in your complete facility, and basis that you identify the equipment. And once the equipment is identified, you start building up the equipment trees, start dividing the components into sub-components, and then do the criticality assessment of the each component. And based on which then you arrive at C3 sheet, which is cause, concern, and countermeasure. And once that is done, you start developing your maintenance strategy. How and where should I how should I attend these parts, components, and how frequently should I attend these parts? And then once this basis this, the RCM reliability center maintenance sheet is developed, which is kind of discussed with team, and it is also discussed mostly with the OEMs also, and to arrive at the final maintenance strategy, which is reviewed and then updated, and again the final preventive maintenance checklist for the particular equipment is rolled out. So this leads to having a proper preventive maintenance checklist, which is progressive in nature, and which also kind of allows you to have a maintenance based on the life of the equipment in the life of the equipment. Please go ahead. Next, please. So this is typically how the equipment tree is made. Like I've just taken a simple example of a bicycle, how it is, kind of the sub components is identified, the seat, the tire, the chain, and all, and basis which you then finally arrive at what are the important components in this this bicycle to really make it run. Next, please. So further talking more on the context of the pharma industries, what I've done is just try to typically take a case of fluid bed dryer, where the fluid bed dryer is divided into the major system, like its spray system, then hot air unit, then chamber, and all those systems are divided, and then spray system is further divided into typically different components. So each of these components are in the equipment tree is taken into consideration. Then we evaluate what are the failure modes in this, and basis which then we arrive at the C3 sheet, as I said. So all these components has to be kindly assessed properly with help of a team, and there has to be a team of engineering projects from user side, and preferably from vendor also, to do a complete assessment of the equipment in depth. Next, please. Now, these are some of the questions which we generally take up during developing the RCM exercise, which is, what are the functions and associated performance standards of the assets and its present operating context, what way it will fail, and it will, how it will fulfill the function, what are the causes of functional failures, what happens when each function failure occurs, and also these are the questions which we generally ask while doing the these are some of the questions we generally ask during while doing the RCM exercise. So this has to be more of an interactive exercise, and done with a cross-functional team. And more detailing you do on the equipment, more your robust the preventive maintenance checklist will be, and thus the equipment can be maintained properly. Next, please. Well, so I think it's very crowded slides. I'm unable to see this. So this is basically our RCM task decision worksheet, where you kind of list down all the components and what are the different failure modes in that component, whether it will have an impact on equipment, or it will have impact on environment, the performance of the equipment, and basis which then you kind of assign whether this should be taken up in the preventive maintenance, or it should go as a part of predictive maintenance, or it should be an autonomous maintenance where the operator himself kind of take it up as a CLITA practice, and attends after the during the setting up of the equipment. So this is a very elaborate sheet which comes after the RCM discussion is taken place, and this then all these learnings and discussions are taken up in your PM checklist, as I said earlier. And next, please. Well, again, so this is the testing practices. Like I said earlier, we have got a thermography camera which checks whether all the terminals are getting heated up or not. Then we have got a ultrasound detection which checks whether there are some air leakages or not. Then there are vibration analyzers. So these are, and then the oil testing is also done in the gearbox to understand if there are some metal particles, whether the viscosity of oil has increased or not. So those all things are generally, kind of always tested. So which, kind of gives you whether the equipment is running towards any failure or not in near future. and you can, so that you can always have a timely check on the equipment and attend or change the part of the equipment if required. Next, please. So now this digitalization is becoming a very important aspect in maintenance. So like computerized maintenance management system, which is primarily enhances the kind of performance of the equipment. And this we have started implementing in couple of, at least I've seen in couple of companies where we have started implementing this, and it's more on the mobile application. And this gives you complete visibility on the performance of the equipment. Obviously, you can assign the task from your mobile itself. You can always manage your spare parts inventory, and basically it goes completely paperless. And from standing in front of equipment, person can always scan the QR code and raise the request for maintenance. The maintenance requests are tracked. So the proper SLA has been, SLA is derived and kind of maintained in this. So this is becoming largely a global practice now, and people are approaching towards this. So this is one of the very important futuristic software which is coming into place. Now, second thing is getting IoT in the maintenance area, which is which will help us in kind of real-time monitoring of equipment like chillers, your boilers, your air compressors, and all that. And of course, it also enhances the safety when you kind of all connect all your hazardous equipment also in reactors, your cold room chambers, and all that, storing the chemicals. And all those parameters are thrown back on the system, and you can always access it from your mobile. And which of course increases the efficiency of the process as well as of the maintenance team also. And all the critical parameters are thus monitored on your system, and it's really it helps in kind of giving the complete visibility to the maintenance engineer and for the plant manager also. The other important thing which has kind of started playing a role is on the augmented reality and the virtual reality, where we kind of digitize all SOPs, and the person can do maintenance of the equipment. While doing the maintenance, he can always access the SOPs, and he can fill up the SOPs by the verbal command. And he can also access the OEMs in case of he's kind of stuck up somewhere while doing performing the maintenance operation. And at the same time, with help of VR, we can always kind of create a training modules for a person to virtually see the equipment, work on the equipment, and kind of understand the equipment better. So the training modules can be created for the engineering team also, and as well as for the operation team also in this. So these are some of the important aspects which I've tried to cover quickly in this presentation, and which helps us to drive and improve the efficiency of maintenance. Yeah, next please. So that's about from my side, and open for any questions, please.
Thank you, sir. I invite Siri sir for the Q&A session.
Thank you, Sanjeev, for a nice presentation. I know you are coming out of dengue just now, and still you came here to give the presentation. Thank you very much for that. I think it was good presentation, very comprehensive. I think there are a few questions. It is actually spill out from the last presentation, but I think that question suits you more. The ducts of the bees and coaters, particularly fluid bed dryers as well as the fluid bed processors and coaters, how the duct cleaning should be done, and focus on the FDF.
Sorry, sir, I just lost your voice. I think there is some disturbance.
Can you hear me now?
Yeah. Now it's better.
Yeah, better. I think the question is from the Deepen Shroff of Dr. Reddy's.
Mm-hmm.
Fluid bed dryers and fluid bed coaters, duct cleaning or duct maintenance is an important criteria which is being focused by the FDA consistently. Are there any best practices there which you would like to tell us?
See, I mean, what I have experienced is that people, they used to open up the ducts and clean the ducts, which is quite a laborious activity, and it takes time also. And then again, it's kind of time consuming. So then the next system came into place is having a troll balls in the ducts. Now it all depends upon how far you want to go into with the cleaning of the ducts. It could be as the OEM defines, it could be 2 meters from the flap wall of the duct, or sometimes people, they go right up to the HEPA filters. Basically, the best practice is, the best way of doing is having a troll ball in the system. It's slightly expensive, but it is much faster where you do the complete WIP of the equipment in place. So you don't have to really open up anything, and with one command, having a WIP skid, you can clean the entire equipment, including the ducts. So that's the best way to do it as of now. People, they are also using Karcher system to put some hose pipes inside the thing, but then it's still not found very effective.
Thank you. Another question is machine learning. Can it be used for the predictive maintenance? Have you seen any application like this?
Well, it can be used. I have not seen any application so far. So when I was talking about AR, so what we were planning to do is, kind of capture some of the abnormalities in the equipment running into the AR platform. So that is being done. It's still working on, and people are working on that. But I have not seen anything practically working anything on that in pharma, at least.
There is one more question from Mr. Nitin Agrawal of Ajanta Pharma. What is AR, VR, and MR technology? Can you explain a little bit?
So AR is basically augmented reality, where you virtually see the equipment. So basically, let me start with the VR. So VR is a virtual reality where you create a virtual area and the equipment. So the person sees the equipment, he can kind of feel the equipment and operate based on the virtual background which is created. So this is more or less used for training of the personals, where he can kind of operate the equipment, he can carry out the maintenance of the equipment, and get trained on the SOPs. So that is the VR. Second thing is AR. AR is where the person can, is augmented reality, where the person sees the equipment. He can also kind of read the SOPs while he is, kind of working on the equipment, and without having a complete visibility of the equipment. So let us say if I'm doing any maintenance of equipment and I'm stuck up somewhere in the gearbox of let us say compression machine. So I can go back on the same module with the wearable glasses, go back to the training system, see how the gearbox has to be opened while I'm actually seeing and performing the operation on the gearbox. So this concept is now picking up, and it is really good. So I've seen, started working on that. We are working here also on this, and then it's picking up basically.
Last one was, what is MR?
Sorry?
What is MR?
Yeah. So MR is mixed reality. Basically, it is a combination of these two, where the person sees a SOP, again he commands. So it's by and large more or less he sees a virtual atmosphere also, as well as the equipment also.
Thank you, Sanjeev. Nice presentation, and we'll wait for next presentation. Thank you.
Thank you.
Thank you, sir. Our last speaker as a part of the IPA Quality Forum subgroups is Mr. Mustaq Sherasia, Senior General Manager, Sun Pharma, who will cover on visual inspections of parenteral products. Welcome, sir.
Yeah, thank you. And good morning, good afternoon, and good evening, all. It's an immense pleasure for me to represent IPA subgroup Forum 4. The important topic which we considered for work together with the 5 companies with ambition to strengthen our processes on visual inspection process. And we develop a guidelines, very detailed way how to perform visual inspections of sterile product with a different dosage form, and that is given a detailed processes based industry practices and some of the good approaches which are considered for visual inspection in summarized in coming slides. So next slides. So this is our team introduction, and we would like to thank you our team members and colleagues from 5 different companies who have participated. And I would like to thank you our mentors, their continuous guidance and supports to make these guidance documents very effective and meaningful. Next slides. So as a IPA subgroup 4, our vision was to create a detailed guideline with a very robust processes and seamless process to provide a defective analysis and visual inspection approaches clearly with the industry views and what other approaches to be applied with alternate practices to be followed while difficult to inspect products are there. So that details are there, and we'll have a look in the coming slides. Next slides. When we started these guidelines, our purpose and scope was to cover the different formulation of the products, which is a liquid injectable products, ophthalmic products, and we also covered a lyophilized and suspension product, which are very difficult to inspect the products. And based on the request to the group, we have also touched upon how to inspect the media fill containers after performing the media fill inspection. The basic concept which we focus on inspection capability and ultimate focus was with respect to patient trees. So that was the prime focus area, and based on that, the entire guideline document has been covered. Next slide. If we understand the inspection process flow, normally the entire filling process completed and then followed by 100% inspection is performed, and then it goes for a supplemental and equal testing. And if it goes successful, then it goes for the packaging process. The two important consideration is the rejects, which is very important to consider after completion of inspection, and their trend analysis and classification of defects, which is very important consideration that we will discuss in the next slides. And second is a supplemental testing, which is very important for complex products and products with a specific container closure configurations, which is very difficult to inspect visibly. So that is covering also in coming slides. The AQL sampling is very important after completion of 100% inspection, and it has to be covered to identify as per NC standards, and sampling plan is to be followed to ensure that you will get the confidence that the inspection which is carried out is effective and as per the pre-established procedure with the qualified inspectors. Sometimes there is a remediation practice is also followed when there is a inspection fails at 100%, then it's recommended based on investigation to perform a re-inspections and followed by statistical AQL sampling. And two-stage inspection is also recommended in case of you have a probability of false failure during your visual inspection. Then the second stage inspection is recommended to identify those false rejection. However, the limit and criteria has to be defined with a well scientific rationale for performing for these two re-inspection and two-stage inspection processes. Next slides. So this is very important slides, and I would like to touch upon more in these slides. While considering the visual inspection of product, the thought should be start from the development stage itself, and that is important consideration to take up during development of the product. And this stage is important to identify the intrinsic particulate which are there from primary components, formulation components, and common source which is there inherent in the product itself. The second important consideration which we need to identify and controls during the production process is the extracting particular which are normally entered during the manufacturing process, and that has to be controlled through a different processes in the manufacturing lines. So the basic topics which important is particulate removal is ensured with a specific processes which respect to a cleaning. So cleaning important with respect to glass container. When you clean your glass containers with a automatic vial washing machine, so machine parameters with their specific ranges has to be ensured during routine operation to ensure that your glass containers are adequately washed and it does not carry further particulate to your final products. And similarly, the container and closures, there are different configurations available in the market. Sometimes it is ready to use and sometimes it is ready to sterilize. So the periodic monitoring of these closures is also considered to check as a part of routine monitoring program to ensure that those closures are free of particles. The important source for glass particles is a depyrogenation process where the probability of breakages of vials are there, and that is carried to a particles to a depyrogenated vial, and then subsequently it goes to a filling. And it is very difficult to identify because it's an integrated process. So consideration has to be established during qualification of depyrogenation tunnel and synchronization of the filling line to ensure that we should avoid the minimal breakages during the depyrogenation and filling processes. The another important is equipment washing, which is ready for the filling component, and this equipment cleaning is a important consideration to prevent the particulate contamination, and specifically the soft floor display on difficult to clean component will help to reduce this, and specific cleaning design considerations like needles and all those components has to be designed to ensure that it is adequately cleaned and there is no probability of further carry forward contaminations from difficult to clean such components. Lastly, the filling line itself is a potential source of contamination and particulates prevention. The components which are directly contact in the product itself, like syringes, vibrator bowls, all those are threading if it is not adequately cleaned and autoclaved and wrapped properly in the environment. So it will lead further particulate contaminations in the product itself. So these are the baseline controls starting from development and then commercial. But important consideration when you do your visual inspection, the trending. The trending of visual defects is very important consideration to understand your source of particle and to identify those particles from the various sources, and fixing the root cause analysis and taking a right corrective action is very important. So typically the trending is done with respect to different defects and total defects, and it is statistically evaluated with a number of loads, and then pre-established alert and action limit are established to investigate in case of you found any of the higher particulate matter during your routine life cycle approach. And it is also recommended to have to evaluate your AQL failures incidences to understand maturity of your visual inspection process. Next slides. So as I mentioned earlier, the unique product containers and consideration is very important for visual inspection, since lyophilized products is having a powder cake, powder formulation is itself powders, and emulsion and suspensions, umber containers and translucent plastic containers and large volume containers and combination products. Those products are very difficult to inspect product, and you cannot directly do a visible particulate matter inspection. So here the special recommendation, and we have covered these things in the detail in our guideline. The alternate inspection strategy to supplement the testing has been carried out, which include transfer of parent products to a different containers, clean, visible containers, filtration technology, retaining the particles and identifying those particles, clarification process, and the sieving process of followed as a part of alternate practice and supplement testing to identify particulate matters in the product itself, which are very difficult to inspect. So interpretation of results is very important with respect to classification. The classification is defined in a critical defects, which is having a serious impact on the patient health, and there are major and minor defects. So each category has to be defined with limits on a scientific rationale, and based on that, if it crosses the limit, the investigation is to be carried out in the details. Next slide. There are three different inspection processes are followed. First is manual inspection process, second is semi-automatic, and third is the automatic inspection processes are there. So we will touch upon all three in coming slides. The manual inspection process is a inspection process which is widely used in the industries as of now. And inspector is pre-qualified prior to start of this manual inspection process. The critical process parameters includes for manual visual inspection activity is light intensity of the inspection booth which is used, and there is a defined criteria for this particular intensity. Backgrounds and the contrast, white film and black film is to be utilized, and there should be a periodic replacement program for this to consistently meet the adequate contrast. The inspection rate is another important consideration. Normally, the 10 seconds per container is allowed, and for each background, it should be minimum 5 seconds. However, considering specific product, this time could be taken more if the product is having a different complexities. The container handling and the movement is a important consideration because since it is a visual inspection manually, the holding of containers varies from 1 ml to 100 ml in a different numbers. So that criteria we have provided in detail in the guidelines, and a team can refer those criteria in detail. Magnification sometimes helps on very small containers like 1 ml and ampoules where we can utilize. The important consideration is inspector fatigue, since all inspectors are human. Constant inspection will lead a false rejection and false count as well. So highly recommended to have a inspectional break for at least 10 minutes every hour. And this can be controlled by a automized technology like auto light off techniques, and some of the techniques which are used is a alarm techniques which can be reminder inspector to break every 1 hour. Next slide. The another visual inspection technique is a semi-automatic technique like manual inspection. It has an advantage of high speeds and automatic movement of material. It does not require to move this material by taking and holding small containers with a sequence. This goes on the conveyor, and machine will automatically detects, and then it has to be operated by qualified operators. The important consideration and the parameters which to be considered is the spin speed and the inspection rates. So both are controlled through a conveyor, and it can be easily qualified and has to be maintained during the routine batch manufacturing inspection process. Next slide. The third inspection technology is automated visual inspection, and this is now emerging in most of the companies and industries. The two methods are used. One is light obstruction methods, and second is imaging methods with a camera. In automatic inspection machine, the advantage is that the machine has having a electronic control, electronic sensors, cameras, recipe-based controls, and auto rejections outputs, and good goes on the container. So, however, there are some of the limitations with the automatic visual inspections, but now a modern technology visual inspector machine is available to detect all type of defects. Next slide. So this is very important technology which is used specifically to detect metallic, glass particle and rubber particles in the powder and lyophilized product. The X-ray machines are useful technology in case of any embedded glass particles or rubber particles are there in the powder and lyophilized formulations. However, special consideration is to be considered while utilizing the X-ray technology with a product development history and establishment of stability consideration to be taken prior to implementation of this technology for specific powder and the lyophilized product. Next slide. So coming to the preparation of the kit prior to qualification of visual inspector is very important consideration, and we need to consider these steps before you qualify your inspector because right kit, right qualification and right size of the particle which is required to be challenged as a part of process to ensure that your inspector are rightly qualified and those are detecting particles during your routine commercial batch. So kit has to be a very controlled environment, and while selecting the kits, it should be defined very specific criteria, like it should be free from particles. The container should be absolutely free from visible particles, and the expiry consideration is also to be defined. Second important consideration is each kit defect should have one category of rejection. It should not be have a multiple category of rejection. Otherwise, it is very difficult for inspector to judge this defect during qualification. And there is a minimum criteria of defect distributions are defined is 5% to 15%. And all these defect has to be covered from critical, major and minor defects. It should not be like that you have only minor defects in the kits and inspectors are qualified. So it has to be equally distributed, and that criteria we have provided in the detail in the guidelines. So practice is used for specifically this size of the particles ranging from 100 micron to 250 micrometers, and fibers it may be ranged from 500 to 2000 fibers. Along with preparation, once you are qualified with inspector, the maintenance of this kit is a important consideration, and this maintenance has to be through a procedural controls on issuance, retrieval, and periodic inspection of this visual inspection kit. And this kit has to be controlled and stored under the lock and key and under quality assurance controls. The kit can be further replaced in case of you found any damage during visual inspection, and it should be replaced with a pre-qualified container. And after expiry, if you find any specific visual defects, then it can be discarded at all. So that has to be controlled for the kit very considerable amounts. Next. So after preparation of kit, the important consideration is to qualify your visual inspector, and it goes in a 3 phase. Phase 1 involves detailed training, work experience and medical checkup, and phase 2 involves a detailed consideration. Inspector goes in the phase 3 training to understand and perform the real challenge test of particulate removal testing and visual inspection during this kit. And this phase is very important because here the important consideration is the inspector is challenged with a previously prepared kit, and it is a challenge to identify all defects which are kept in the visual inspection kits. The important criteria here to be followed is distance criteria, handling of containers. It could be from 1 to 5, depends on the size of the containers which are considered for inspection, and total time of visual inspection taken. Fatigueness of the operator is also to be part of your visual inspector qualification. Lighting criterias and the bracketing approach of container is also considered while you qualifying your inspector, and the environmental conditions which you need to consider during your qualification of the inspector. Next slide. The another aspect which we considered is media fill containers inspection. Like the manual visual inspection process, media fill inspector is also to be qualified. However, here the specific consideration is given to identify containers which are breaching container and closure integrity during the prior to incubation inspection, and on 7 and 14 days to identify turbid containers which are there if it is found during the inspection. So here also similar approach when you prepare the kits and qualify the inspector, expiry of the kit has to be defined. Inspector has to be pre-challenged with a physical defects and turbid containers. And the special recommendation here is to consider color indicating media, which is a very useful technology available to utilize and easily detect the contamination. So it is easy for visual inspector to detect any contamination which is there in the media fill itself. Next slide. The second part after media fill inspection is a control sample inspection. It is very important consideration which is need to consider as a part of inspection program. Typically, this is carried out annually, and it has to be carried out by qualified inspector, and criteria has to follow as per manual visual inspection. There is a specific consideration we need to consider for here also a destructive testing for live powder suspension, and which is to be do as exactly like we do for supplemental testing for identified vial, and SOP should be defined very clearly to perform the destructive test in the control sample as well. And the last is important consideration in the control sample inspection is to consider adequate reconciliation and the documentation of the inspection which is carried out. So it gives a full traceability of inspection which is performed as a part of annual program. Next. So this is very important consideration which we considered in our guideline to cover the investigation consideration of visible particle, which is very challenging to identify the source of particle. So we derived some of the common points which is mentioned in our guideline with respect to the first important criteria is to understand when you found out of limit visible particles, the triggering of the investigation and understanding the type of defects which are observed more. So the type of defects is very crucial stage and trigger point to understand where you need to focus in the investigation. Let's say you find a higher glass particle in the inspection process. So your focus would be more on the glass containers, breakages, probability of source from the vendor itself, and any adequate breakdowns or any abnormal findings during the processes. So that is an important consideration. And if you do not have any specific visible particles, which is very difficult to identify, then identification technology has to be considered with a analytical processes like morphology, IR, scanning electron microscopy, X-ray, and elemental analysis will help to identify you the particles and their source and deriving a conclusion near to your source, and you can put a adequate corrective and preventive action. So here I would like again to emphasize on specifically trending of defects, which is very important steps which we need to consider to identify our defect patterns, and based on that continuous process verification program will really help to focus on where to investigate as a pre-alert limit, which is out of trend. And if you cross that limit, you can save your batches at early indicator, and you can focus on the investigation processes. The filling line, when you are evaluating one of inspection process and total out of limit processes, the filling line consideration is to be considered whether it is product specific higher rejection or filling line itself is contributing factors, and it is observed in all the products which are manufactured in that particular line. So it will easy to detect you and identify the source of those particles, and accordingly you can fix your filling line issue if it any there. Last but not least, in case of specific higher critical defects, health hazard evaluation is required to identify the patient risk before you take any disposition decisions or any decisions on the release of the batches. And additionally, sometimes supplemental testing also helps to identify source of particle and consider your investigation process. So next slide. Thank you, and I'm ready to hear any questions from the team.
Thank you, sir. I invite Shirish sir for the Q&A.
Sir, thank you for the nice presentation. I think your group was the first group to come out with the guideline, I think, in all the 4 groups.
Yes,
it was
sir.
very comprehensive document, very nice document. There are few questions. First question is related to the inspection process itself. Is it necessary that inspection should be carried out as soon as the filling is done, or we should wait for the filling, spend some time, more days, and then do the inspection, so that chances of any particles are increasing after some time? So what's your take on that?
So here I would recommend, if you have experience like that, if you have a continuous line with a automatic inspection, and normally industry does immediately after filling, and sometimes there are offline inspection also. So it is both immediate and after sometimes. But if you have any experience specific to a product, which is developing over a period of time. So during development study, that has to be considered, and you can define your timeline for inspecting those products.
Thank you. Second question is related to the manual and semi-automatic inspection. In both the cases, the inspection output will be limited by the 10 seconds criteria. Each vial has to go for 10-second inspection. So in a day, you can do maximum 2,400 vials per person, right?
Yes.
If you 10 seconds, then per minute, 6 vials, and then if you look at the 400 minutes, it comes to be 2,400. So that's the limiting factor, whether you use semi-automatic or automatic, semi-automatic or the manual. Automatic is totally different.
Correct.
That's what, okay. So that's actually limiting factor to the whole thing. And the number of people need to be qualified are very high in such cases.
Yeah, only advantage would be the fatigueness part, where the inspector has to pick up the container, keeping on specific inspection booth. So those activities can be eliminated, and inspector will have a comparatively relaxed time and fatigue compared to the complete manual inspection program.
Thank you, Mustaq. Thank you very much. No more questions. Thank you.
Thank you. Thank you.
Thank you, sir. It's great to see, it's wonderful to see how much time and effort has been invested by each of the subgroups. It's a great example of collaboration and teamwork between the companies. So with this, we come to the end of the first half of the session. We'll take a 15-minute break, and we'll resume at 6:15, where we'll have a panel discussion, and then we'll close out for the day. So let's be back at 6:15. See you then. and welcome back, everybody. Do what is written and write what you do. This is known as compliance, and we all work within this framework. And we tend to, we try to achieve this and sustain this. So this brings us to our next discussion, a panel discussion on GMP reflections on the journey so far. I invite the moderators, Mr. Vishnukant Pitti, Associate Partner, McKinsey & Co., Mr. Sirish Belapure, Senior Technical Advisor, IPA. Joining them will be the panelists, Mr. Vipul Doshi, President, Corporate Quality Assurance, Zydus Cadila, Dr. Ranjana Pathak, President, Global Quality Cipla, Mr. Indrajit Bose, Global Head of Quality, Dr. Reddy's Laboratories, is not there today. Dr. Rajiv Desai, Global Head of Quality Lupin, Ms. Jila Breeze, Global Head of Quality and Compliance Sun Pharma, and Mr. Dilkesh Shah, Vice President, Quality Torrent Pharma. We welcome you all.
Hi, everyone. A very warm welcome to my fellow panelists in this session today, which is the concluding session of the IPA GMP event. We'll actually be focusing on reflecting on the kind of trajectory that the industry has been on in terms of ensuring and assuring good manufacturing process practices, or GMP. But more importantly, we hope to have a meaningful discussion and debate around what the future holds, or the future of GMP, if you will. To get us started, I think GMP as a term is something that is widely recognized globally. We all know how GMP came to be as a concept. The realization that endpoint quality testing is potentially not sufficient in itself to ensure the kind of quality excellence that patients need and deserve globally. Right? In fact, it's important to ensure that quality practices, good manufacturing practices, are embedded into day-to-day operations on the lab, in the shop floor, etc. Over the last decade, we very well know our understanding awareness globally has been evolving and has been increasing over the period of time through regulations, guidelines, whether it's the ICH Q7, whether it's the ECGMP, whether it's the USCGMP, you name it. In fact, over the last couple of days itself, we heard some of the speakers talk about some of the future forward themes that are likely to shape how we think about GMP going forward. The increased adoption of digital analytics, automation in quality, and the kind of impact it can have on quality outcomes. In today's discussion, I would love to explore some of these themes, these future forward themes with you in a little more detail, and get some of your thoughts and perspectives around this over the next hour and a half. But before we get into the depth of that, just to get us started, I would love to go around the room and just get very starting thoughts, just quickly, on what defines GMP for you so far, and what defines GMP in your opinion as you think about the future of GMP for the industry. Maybe Rajiv, you can get us started, and I would love to go around the room thereafter.
Thank you so much, and it's a pleasure to be back on the IPA forum. And with all my colleagues over here, we've been talking to each other several times earlier. But yes, I would say GMP, I would still extend it as a CGMP, current good manufacturing practices. So I love that term. And this is something that I would definitely try to elaborate. One is it's more of consistency of the products that you manufacture. It's also about cost effectiveness in terms of your manufacturing processes, compliant to the regulatory guidelines. That's so important that every time you come out with some new guidelines from the agencies, you should be able to make sure that your product, your processes are in line with those guidelines. And most important, maintaining the entire quality from the entire value chain of the process throughout the manufacturing, and that's something that should be ensured, and that's what I feel the entire CGMP is all about. Thank you.
Thanks so much, Rajiv, for sharing your thoughts. Jila, would be great to get your reactions as well.
Sure. Can you hear me?
Yes, Jila.
So good morning and good afternoon to all of you guys. So first, allow me to thank IPA Group for setting up a great 3 days GMP training. And thank you for all the presenters. Valuable knowledge. So to me, to simplify it, GMP is applying continuous learning. And the past 3 days IPA, it was a great learning. So understanding end-to-end of our processes, from the material to manufacturing to release, and a post data analysis of our product, with understanding that what is the intended use of those products, and complying with our regulatory commitments, obviously, with whatever we have submitted to our agencies, our partners, and also making sure our internal established processes are followed with the high integrity, intention of what we commit, with a high integrity and good scientific data understanding of our products and processes, to make sure that we continuously improve our products, processes for end users. Remember, in our mind, GMP is intended for patients. So we shall never forget who is the end users, which is you, me, and our families. And with that, we really need to continuously keep improving our capabilities, mentoring, talents, people, training, monitoring, and continuously learn and to improve whatever that we have in our processes. So that is a kind of like a version of my GMP: high integrity, good people and talents, and continuous improvement in whatever we do.
Great. Thanks for that, Jila. Vipul, would you like to also add to what Rajiv and Jila shared?
Yeah, certainly. And thank you, everybody, and thank you, IPA, for inviting, and good morning, good evening. I will reflect GMP in the same way like Raju and Zila mentioned, but for something philosophically, what I believe in GMP is no harm to the patient. So it's a philosophically patient compliance statement that there is no harm to the patient by the GMP. It also encompasses, Zila mentioned, Raju mentioned, according to me, there will be 5 P and T, like people, process, premises, product, procedure, and technology. So these encompass the GMP. Like what is the meaning of people? Like people, the adequate people, trained, having required skill set people, correct people are correct place, having effective governance structure. In the processes, effective process, easy to follow process, simplified process, and processes required for the activities which are well documented. The premises in the GMP, what I thought and my thinking and philosophy, like a well thought premises, well thought design, well thought equipment in place for stable process for manufacture the product, and then manufacture the product for the sustained basis. Then coming to the product, the robust product to deliver consistent quality to meet the patient need. And then coming to the technology, enhancement for continuous improvement to achieve desired quality with less issues. So these are some 5 P and T encompass the GMP to have the patient compliance in mind. That is what I think my definition of GMP.
Okay, thank you for that. Ranjana, any thoughts from you?
So I will add to what Sheila, Rajiv, and Vipul have already said. And I too agree. I like the C word. And I think it's important for all of us to remember it's good manufacturing practice. Practice means we never get perfect. we have to keep on practicing because the rules get upgraded, technology gets upgraded, people get upgraded, our medicines change, diseases change. So it's a practice that we have to remain current continuously. It's not latent, it's not static, it's dynamic. And in addition to everything that my very highly esteemed colleague said, I would also like to add the provider to the P's. So it's people, process, product, patient, provider, meaning the insurance companies, and the physicians. As we know, things are getting so tight for us that the provider has a huge say to which company is GMP compliant, which product is GMP compliant, and which is not. And we didn't think as much of the provider. Our physicians play a huge role in how we practice GMP on a daily basis. So I think it's a topic in itself. It's huge, it's wide, it's deep like the ocean, and it's not static. And I'm happy that we are discussing this. And Vishnu, I'm happy that was your first question because that is our foundation. Thank you.
Okay, thank you for sharing, Ranjana. Maybe to round it up, I know Indrajit couldn't be with us today. Dilkesh, would you like to round up also? Share your thoughts on what GMP means for you. How would you look at the future of GMP? Dilkesh, I think we're finding some trouble hearing you.
Dilkesh, please unmute yourself. You're not able to hear him, right? Let them clarify, let them rectify the situation. I think we should move to the next question.
Great. I think maybe Dilkesh, we will come back once we've resolved some technical challenges with your audio. The team is just getting in touch. But maybe let's move forward. Just to round up, some of the things that I heard was ensuring consistency in practices as well as outcomes, ensuring leveraging data. We spoke about patient centricity or end user focus. We also spoke about broadening that to physicians, to providers. We spoke about learning and capabilities and talent. I also heard product and maybe adding from my end process robustness. And more importantly, that this is a journey. There is not an endpoint in itself, that we need to continuously keep learning, keep evolving, and raising the bar. Thank you for sharing that, everyone. I think that it sets the tone for this discussion beautifully. Maybe to take this forward, Belapaji, maybe you can share some thoughts of how you think about GMP and maybe take it forward with the first few questions for the group.
I think before going into the future GMP, let us step back and look at it, what has been done so far by the pharma industry overall. You may be aware that before 1970, we were totally import dependent. All the products were coming from the outside and only Ayurvedic products were available in India probably. From that, now we are self-sufficient, not only for our Indian industry, but also we are supplying it to the international market. Currently, India is known as a pharmacy of the world. That I think everybody, it is most touted phrase in our thinking. It contributes more than 20% in volume as well as in the value to the global generics market. And they also say that every third product manufacture you consumed in US is coming from India. So it shows how much we are exporting it to the US probably. Or it is also said that number of US FDA approved plants in India are the second highest after the US. Number of plants approved are in India. More than 600 plants are approved in India by US FDA. More than 1400 by WHO GMP and more than 250 by EDQM approved plants. So you can see how much progress we have done from the pre-independence days. Though it is all looks very nice, but recently last year, you might have seen the presentation by the US FDA investigator on the number of plants which are doing well in the FDA inspections. So India stands at something like 83% acceptable range. Though it looks very nice, number looks very nice. 83% is not bad in any case. If somebody gets 83% marks, it's very good. But it's not adequate because we have to compare with others. And if you compare with the others, China is at 86% better than us. Then the USA is something like 93%, while Europe is at 98%. So it also shows how much we have to go forward over the period in doing this over the period. So I have got two questions for my friends here. I will ask first to the Vipul Doshi and the same question for the Dilkesh. If he is able to speak, I am not sure whether he can do this. So question is, as you reflect back on your own journey over the last 3 to 5 years, Vipul, how would you interpret the progress made by Indian pharma on GMP over the last several years and the success that the industry has seen globally? That's the first question. And specifically, what are the one or two bigger achievements personally, which are your personal and which you are proud of those personally? What more, in your opinion, needs to be done in this case? Vipul, over to you, Vipul. People, are you talking? We are not able to hear you.
Yeah.
Thank you, Mr. Billapure. And then I echoed about the Indian pharma company journey so far. The statistics, what you mentioned, is very interesting, and it speaks about itself. Like the Indian pharma major player globally contribute more than 20% volume and value, and having highest number of US FDA approved site outside US, and more than 1400 WHO approved site, and more than 250 EU approved sites. So the journey speaks about itself. But however, I will give more little insight about what we as a company, along with the government and the other stakeholders. So this industry, with support from the government and other stakeholders, has built a sustainable foundation for success, especially distinctive capabilities in the key area of the value chains. In particular, strong manufacturing, product development, process innovation capabilities have enabled Indian companies to drive significant efficiencies. Over the last 2 decades, the industry has invested heavily in world-class manufacturing facility. Like I'm proud to say that when I visited many of the plants outside India, some of the plants or many of the plants which I've seen in India, I think it is much better than a world-class facility. The industry has significantly expanded capabilities across dosage forms and technology and increasingly focus on the innovations. So I will give an example. In 70s, we have started for the as an API manufacturing. Then we have moved forward to the next level for the strong capability for the dosage form for the OSD. Then we have moved forward for the complex generics and NDDS. And now we are focusing on the large molecule like biologics and NCs. So this is a journey so far what we are looking at as a Indian pharma companies. Companies has taken journey to develop the quality culture embedded in their system and championed by the leaderships. So we see that IPA, like 6 companies, the leaders, they've come together and created this forum. Same way, the leadership of the different companies are also as champion the building the quality culture. There are currently some of the key challenges are addressed in the industries, like building a robust data reliability system for leveraging technology. Like you are aware about that data reliability, we are being hit very badly by the FDA, and then Indian companies has done remarkably well. And then many of the, if you see the observations, there are no data reliability issues. Understanding, upgrading the system and capability in investigation. So that was another area where we were the FDA's focus and we were deep on those. Then strengthening the management system to manage quality, so governance structures have been improved. Embedding the quality into the product design. So we must have heard about the QBDs and many other aspects about the GMP. So now the quality by designs are embedded into the systems. Building a culture of quality across the organization, I have mentioned that, and expanding and upskilling the quality and operator talent pools. So these are the few things which I emphasize on the journey of the Indian pharma company so far. Now coming to the next question of Mr. Bilapore, what I am proud of. So let me tell you that I am proud of my Indian industries, the journey which is done by together, by virtue, by individual. But as me as a head of the quality in my company, one or two things which I am really proud of, that one is the automation. Like I'm very proud that for last 4 years, we have taken the journey for the automation and especially focus on QC first. And now currently my QC is almost 90% paperless. So what we have done, we have fully integrated digital tools like LIMS, chromatographic data management systems, chromatographic and non-chromatographic electronic signatures, fully automated data backup systems, predictive analysis integrated with SAP, and reviewer-friendly environment which we have created. This is not all in the automation is left. We have gone with the e-log books. We have done with the barcode scanners. We have gone with the paper on glass for the remaining activity. So if you see hardly the papers which we can see in my laboratory. Another thing, we have taken the huge work which has happened and which is happening, move towards the Pharma 4. Pharma 4, like implementation of CGMP software across the quality activities. And we are very, very aggressively working towards the integration for the data analytics driven culture and improvement compliance and operational excellence. I will give you an example where I'm very proud of. We have created a smart software. Now, what is the smart software? It is nothing but the historical data and machine learnings are been captured into the systems. And then it is put in our GMP software like TrackWise into drop downs. So now similar type of investigations, so deviation somebody has to do, then they have the knowledge base ready and then they use the database. So this is really help to have individual dependency and then the unproperly written GMP document. So these are the some improvements which we have done. Another thing which I'm very proud of, the paperless environment in microbiology. So what we have done in this, like we have gone with the MODA. I think everybody is doing the MODA, but here the MODA is integrated with my colony counter. So the colony counter is integrated MODA. The earlier days, the microbiologists used to do the reading manually, and then another microbiologist used to do the checking of the plates. And then you're aware that the big plant like what we have, the more than 5,000 plates have to be reviewed on a daily basis, which is another task and impossible to do, and that is very complicated. So now our integration has really helped. One is in the compliance, one is the productivity. And then the microbiologists are free to do much better work into Naval. So these are the few things which I'm really proud of in the automation. And then lastly, we have built the QUEST program, which is quality enhancement through sustainable transformation. So that journey is started with the mechanism itself in the 2016. And then now it is 5 years, we have completed 26 sites. And these programs are on a day-to-day basis going on and then covered almost 100% workforce, which is there onto the floor. So these are the few things which I'm very proud of, which I want to mention. Thank you.
Thank you, Vipul. Are you able to speak? You are able to.
Dilkesh, I don't think we're able to hear you even now.
There seems to be a problem.
I think the IPA team is just messaging me. Dilkesh, if do you want to try disconnecting your headset and seeing if that works? No. All right, everyone. I think maybe Dilkesh, apologies for this, but maybe circle back. And I've just notified the IPA team to get in touch with you to solve the technical glitches with your audio. Maybe I think just getting your overall views on GMP, specifically also reflections on the journey so far. I'll circle back to you on when we get back, right? But maybe just to take this forward in the interest of time, some interesting themes that I picked up from this was focus on culture and capabilities are something that we should feel really good about in terms of the GMP journey so far. Moving towards more complex, potentially more innovative platforms. Spoke about complex technologies, NDDS, etc. And also maybe early forays into how we're thinking about data, new technology, automation, data systems, smart softwares, etc. I think those are the few things that I heard are important as the group thinks about the journey over the last 5 to 10 years. That was very insightful. Thank you for sharing. Now maybe let me pivot to talking about what the future of GMP or how GMP is likely to evolve going forward. As we think about that, it is very important to first maybe talk about how the adoption of quality by design or QBD is shaping up across organizations. QBD, as you know, everyone, is not a new concept. It's been there over the years. The industry has been evolving, is being steered towards quality by design, almost moving from quality by testing traditionally towards a more holistic quality by design practices, systems, organization-wide approach. And even whether you talk about regulations, regulatory focus, guidelines, etc., the acceptance and understanding of QBD also is evolving, and it's potentially one of the first things to talk about when you talk about the evolution of GMP. I do know that all of you are thinking about shaping this or are already shaping your vision for quality by design. But I would love to actually hear about, for some of you, the kind of milestones that you've done, how you're thinking about that vision. And in that context, I have two questions. Maybe I'll start with Jila and then Ranjana, I would love to hear your thoughts on this. The two questions are, one, what is your vision as you think of the industry overall? What is your vision of adoption of QBD for the industry overall, and where do you think the industry is right now? How far are we? How close are we to that vision? And secondly, to achieve the vision in its entirety, what do you think are the biggest one or two priorities for the industry, for your respective organizations towards complete QBD adoption? Would love to hear your thoughts on this. Jila, over to you.
Thanks. I think that this is an excellent question to have a discussion. And of course, it may take days to really delve into this. But as you mentioned, the QBD concept and a PAT, the process and analytical technology, it is many, many, many years old. Maybe I'm aging myself. Almost 20 plus years ago, I started this, understanding this as a young quality personnel with Ejaz Hossein, Ali Afnan, those really the beginner in the FDA who brought the concept of QBD impact. And one of my old companies, we actually were part of working with the FDA at a time to understand the new concept. But let's understand what is really the QBD or PAT intended for. So really, basically, we're talking about creation of a mechanism to design, analyze, and control, right, your processes through a measuring a critical process parameters. And then based on that understanding of design concept, which is different for every company and every product, that is very important for us to understand that I cannot give you a template for 100 companies just to follow it. You really, the whole process of a QBD and PAT is understanding your product, your processes, your equipment. So first is complete knowledge of what you're working with. And I start obviously with all the excipients, APIs, your equipments, what you have validated, what is the input and output looks like. And obviously, post-marketing, continuous improvement based on not only the 3 validation data or 10 first products that you produce, continuously really measuring your execution, because as Ranjana said, life is full of changes. Something that I designed and I produce in a generic world or any pharma world 20 years ago, probably it is not going to look from the equipment, from your talents and people, from your management and leadership, from the regulatory concept, is continuously changing. So that's about understanding what is really QBD. Now, originally, many years ago, when I started learning about QBD, it was kind of like very boxy. You had to start from beginning to the end. But I think that even has evolved. Currently, the way that we at least look at QBD, you can design and understand segment of your processes or segment and redesign quality into it, right? So let me just now talk about Sun Pharma. I'm very, very proud to say that our management is very much involved in this continuous improvement. They really believe in it. Approximately about 4 years ago, we knew that to bring this concept into our company, we need some type of understanding and training. So many in the industry are aware this is not a confidential. We have partnered with a bunch of MIT professors, which are most likely, they are not GMP expert. That's not what we were looking for. We were looking for the understanding and designing how to utilize our output analytical data, and also with understanding our critical parameter, PPK, CPK, CQA, whatever wordings you wanna, and our metrics that we have to understand the real issues that is causing continuous investigation, continuous OOSs. So we started that, and when we started that, we understood very clearly that you need to create a group and a talent internally, because you can't just work with 10 external parties. They don't understand your product, they don't know your processes, they don't know your culture, and so forth. So the first thing, it was dedicated. So we created the groups, we created a goal, right? Percentage wise, what do we want to do? We created designated group, which we call it COE.
I'm in meeting, sorry.
You want to bring the look at it?
Yeah.
I'm sorry. Okay. Okay. By creation of those certified personnel, and that was cross-functional, creating the goal and understanding as what our goals are, then creation of a metric, and then monitoring that as on a monthly basis for the past 4 years. Every 4 to 6 weeks, our CEO, our MD, gets on a call for 3 hours for this program. So you create a culture of importance from a top down, right? And giving the people the talents and training. And plus, we dedicated these people because you can't expect individuals who are doing IPQA in a daytime, in a nighttime, to start working on your process improvement. So dedication is very important. And then rewarding those people, and obviously creating this culture, and expanding it, and continuously learning from that process. That was from a product robustness, and we have started with our actually APIs. Now, in the past year or so, we initiated this QBD or PAT or whatever you want to do on our analytical methods, with the view that why do we have so many invalidated OSs? That is a conversation for another meeting for 2 days, right? And really understanding what is the invalidated OS comes from, because human error has a reason for human error. And we have found out, and we have seen in the past 12 months, a number of invalidated OS has decreased by almost 30%. Now, to begin with, it wasn't that big, right? Although the way that FDA has described invalidated OS numbers sometimes scare people when you see 80%, right? Or 50%, because if you have 2 invalidated OS versus 4, that is your 50%. That sounds huge, but per number of samples may not be huge. But regardless, we have initiated the QBD or a PAT for analytical method recently. So I know that probably I didn't cover all of the execution planning, but I'll be more than happy to answer more questions, because I am very close to this project myself. And I review with all the sites, all the COVs, and our consultant group on a monthly basis to monitor this program. And I have learned a lot of things myself as a black belt. So turning to Ranjana, so maybe you can add your experience.
Very good. So Vishnu, what I love about this is, we're all colleagues, and we're all doing the same thing. Our paychecks have a different name on it, but basically we're all doing the same thing. So I'll pick up where Gila left. So quality by design, as Gila mentioned, is an old concept. I too, back in early 90s, attended lots of Ajas Hussain. He was like the only game in town with QBD. But today it's different. And QBD, in my opinion, has 2 views. One is a narrow, very narrow view, which is, your CMA, your critical material attribute, your critical process parameter, your critical quality attributes, all of those things. And all of those things are important to study during R&D. Because at the end of the day, we heard many of my colleagues say, what is it all about? It's about having good quality product at the right time, in the right place, on the patient's shelves. And all of us have this fiduciary responsibility not to go on back order, right? And how do we prevent that? We prevent that by having consistent supply. In order to have consistent supply, we must know our process very, very well. And we all have, I would say, we separate out into legacy products and into new R&D products. New R&D products, I mean, it's high tech, it's complex generics, we know a lot more today than we knew 20 years ago. And I think it's clear in, I would say, most of our companies in the industry where we have this angst with the legacy products. because it wasn't studied with the principles of quality by design. But today it's different. There's lots of data on the FDA website if you want to go. In fact, there's a whole application and ANDA that somebody has kindly put 170 pages. And if you want to know all the details and how to do it step by step, I encourage you to go to the FDA website, and you will see where this company has studied, and that's what QBD is about, is all your process parameters. What is my blended type? What is the order of addition? What is all the details? Then you go on to your timing, your RPM, your mill type, so on and so forth. I'm actually looking at this document and just quickly verbalizing what I see, but I want the listeners to know that this is available on the FDA website. It's QBD IR tablet module 3. Extremely helpful if you want to go and introduce this in your own company. And then you understand what are the material attributes of the input material. And my friends, it's extremely important for us to understand this and understand the process steps. And at the end of the day, quality attributes is what we measure. Is my content uniformity okay? Is my blend uniformity okay? Is my assay okay? Because all this will be okay only if I have studied the individual parameters, whether it be your glidant, your lubricant. API we all study, we're all champions at studying the API, but there is a lot more to study besides the API. So one is forward-looking, which is the new R&D products, and then the other one, unfortunately, we all have to deal with is the legacy products. Now, if you're going to say, how do I deal with the legacy products? Look at, and I think Sheila touched upon it, is look at your process capabilities, look at your trends. You have a significant body of data. And can I look at individual parameters and look to see where is my process not as robust as it can be? So connect the lab to the production flow. And I think it's important to also mention is in our own company, we should not be looking at lab incidents separate from the manufacturing incidents. If I'm getting failures in a product, I must go, and I know everybody does that OS investigation and all of that, but I think we look at things in silos. What I want to encourage all of us is look at the holistic view, bring it all together. Even though something is validated, 3 batches, excellent, Tali. 10th batch, 20th batch, that's our reality. And we have to build for this reality because we may have control over our equipment, maybe our people, but we don't have so much control on the input material. Even though you have a good supplier certification program, I bet you there are some things that your vendor does that we don't know about. They've changed. They've changed the site, they've changed particle size, they've changed something, they've changed surface area. And our tests are not so discriminatory. So I think quality by design is at the heart of our operation. And none of us can do enough to study our process. And it's a continuous process. It's, as Rajiv said, the C, as I mentioned, is the practice. And in between, all my colleagues have said it very well. And Vishnu, you have to stop me because I could keep talking about QBD. I mean, there are seminars for QBD for days, right? And you want us to summarize this whole holistic approach to product robustness using QBD as a tool and all the fancy words, CPP and CQA. You can make your own acronyms. But at the end of the day, my friends, it's what's my input and what's my output. We don't want recalls. We don't want FARs. We do them if we have to. But can we build our process in such a manner that the product will meet the last date of the expiry date period? Vishnu, thank you for allowing me to speak. And you can tell me if I have more time, less time, you can give me the cues. When we were physically there, somebody would show the 5 minutes, 2 minutes, or the yellow light would go on. So we have to rely on Mr. Bilapre, you and Vishnu to tell us with regards to time because we are so excited about our topic. Thanks for listening.
I think we're good on time, and thanks so much for sharing. I took away a lot, actually, and as you rightly said, it's not enough to spend 5 minutes just talking about QBD. It's a vast topic. I picked around themes around designing, and design thinking, analyzing the right data, control systems. How do you work towards it? There's an element around culture that you build, the kind of dedication, focus around it. Ranjana, you also spoke about splitting it into also looking at not just R&D involvement for all the new products or relatively closer to home products, but also looking at what you called as legacy products. So thank you, Jila, Ranjana, for sharing this. I know this is, I think, possibly like 10%, 1% of what you would want to say, and so limiting yourself in the interest of time. So thank you for sharing at least that. Just one quick check, I think, Dilkesh, do we have you back?
Am I audible, Vishnu?
Big applaud for Dilkesh.
Okay. Sorry for apologize for this problem. Actually, I am somewhere in the hotel, so could not able to have a proper connection over here.
Not at all, Dilkesh. So now let me put you into the spot on the spot, right?
Sure.
Good if you could share thoughts on what defines GMP for you, and if you just reflect on your journey over the last decade or so, right? What are the 2-3 things that you're most proud of? What do you think defines the journey going forward? If you just think of GMP, and we've also spoken now about
Sure.
QBD, thoughts on that would be helpful, right? So over to you.
Sure, Vishnu. Thank you so much, Vishnu. And thank you for arranging this panel discussion, and thank you IPA for inviting me for this panel discussion. So your first question was what GMP means for today and where you see in the coming future. So I just briefly tell that as per my understanding, what GMP is the designing your facility, system and processing into the manufacturing and processing of pharmaceutical product and devices in a way to ensure the consistency of the quality of the end product, and not only for a few batches or few product, but for all product in a sustainable way that ensure both acceptability of the patient and the efficacy and safety of the product. So this is the concept which I believe is for GMP, and you see the life cycle of I mean the conceptualization differences from the decades in the 2000 to current situation. There are a lot of drifting into the automation and processing. So GMP is the applicability of practices which I see is that it is from the conventional unit stage wise batch manufacturing or end testing concept to semi-continuous or continuous manufacturing and real-time testing approach. So there is a drifting on the processes, and that is how the applicability of GMP we need to think in a different way. That is what I see into the future. So going forward, what I believe is that the process and GMP system needs to be much more sustainable, and believe that that can be definitely achieved by a lot of new technologies. There are a lot of advancement of the use of PET tools which can avoid the QC burden, QC testing, QC failures. We can utilize a lot of IT platforms and applications for digitalization and real-time data analysis. And of course, these are the new challenges, and that has to be aligned with the GMP expectation, because moment you shift to those kind of a new platform and areas, you also need to think that how it will be consistently remain into the compliant state throughout its life cycle. So a lot of computer system validation would come in picture in the future for the GMP application. So though I believe that initial phase, this is going to be a very challenging task, but end of the day, I think it result in the sustainable environment and also helps in the real-time decisions for the batch release. Moving forward, I would consider there are several elements of the GMP which would be continuously adopting. For example, do right first time approach by applying the what Ranjana, Jila, and everybody says that the quality by design approach, both in not in only processes but also into the analysis, making your processes robust so that you will have a minimum failure, minimum recurrence of the issues, minimum deviation and event, and that is how you can get rid of the investigation of those aspects. So the another aspect which I feel that in future would be helpful from the GMP perspective is the data-centric decision processes and improvement in the process based on the real-time data analysis. And that would also build in the assessment of the quality metrics and quality indicator to understand the maturity of the quality system. And I believe that improvement based on all these aspects would definitely help industry to sustain the product robustness and the GMP compliance. And one another aspect which it's migrating is that the industry is looking at the reducing the human intervention in the processes by using various applications, and that is how we can enhance our consistency of the processes and applying the risk-based approach, and that would really result in the maturing the quality system and ultimately enhance the compliance for the future. So this is what my thought on the GMP for today and what future I see.
Great,
Now,
thanks.
another
Sorry, go ahead.
Sorry.
Please go ahead. Please go ahead.
So another question you had on the reflection of the journey so far, and what are the progress made by Indian pharma on the GMP aspect. So I would say India has made significant progress in over last couple of years in aligning expectation of both good manufacturing practices and quality management system. Though there were a lot of old guidelines, but there are not a lot of new thinking and concept in terms of updating the regulatory environment and the guideline that includes quality by design approach, applying ICH Q9, Q10 pharmaceutical quality system, and that has to be built in throughout the life cycle of the processes and the product. So ultimate goal is to make the processes more robust. So I would say the pace of evaluation of quality management system of the pharma companies, particularly in India, is much faster in last 5 years. And I think that is ultimately getting strengthened the overall control and the processes into the product designing and the commercial aspect. One aspect which I feel that as an industry or as a company we have come out is that we have done a lot of improvement on the data reliability aspect by enhancing the quality culture, or I would say by enhancing the overall approach by applying the Part 11 compliance and upgrading the legacy instruments so that the data security and data protections are in place. So I would say that as an Indian industry, particularly into the regulatory environment, as an industry, we have done a lot effort and lot challenges we have taken, and ultimately we are in the very comfort zone, and that is evident by a lot of if you see last 2 to 3 or 483s or maybe regulatory observations, those are mainly I would say on the investigation aspects and not on the data reliability or the integration aspects. But yeah, there is a long way to go because GMP is always a CGMP, so it is a continuous journey. And by adopting the relatively new concept of continuous manufacturing, automation, digitalization, will end up with new challenges for the GMP. And at the same time, it would uplift the overall compliance status. Another aspect is like India is a bunch of, or it's a mix of small scale industry, middle scale industry, and large scale industry. There is both regulatory environment as well as we have a different markets, different laws. So aligning all regulatory standards and ensuring that the Indian GMP standards are also up to the global level, that could be one of the area wherein IPA as an industry we should consider. And as maybe Sheila and Vipul Bhai has told that investigation improvement is again an ongoing process, and if you want to have a sustainable enhancement, then we should really focus on applying various root cause analysis investigation tools so that we ensure that we are really achieving the robust processes wherever there is a room of improvement from the system perspective or the process perspective or from the product perspective. We can take appropriate kappa and ultimately reduce the failures both in terms of batch investigation or in terms of any events. So this is what my thought on these two aspects.
Perfect. Great. Thanks so much, Nilkesh, for sharing this. I'll actually pick out one thing that you mentioned, right? You also spoke about the kind of challenges that are important to for the industry to consider when we think about quality by design, GMP of the future, etc. Right? Maybe I'll hand over on this particular piece around how do you think about complexities that are inherent when you think about shaping the future. Maybe I'll hand over to Birapaji to at least take that forward.
Thank you for sharing the vision and journey so far. However, there are many structural challenges which, when we are implementing the QVD or the GMP, we are facing all the time. And few of them are known to us. We have got a very diverse portfolio, having lot of SKUs, number of SKUs. I think every company has got SKUs from 500 to, I don't know, 3,000 or 5,000 SKUs everybody is producing. And different kind of the products, dosages, as well as complexity in terms of the geography. We supply it to the non-regulated countries, regulated countries, semi-regulated countries. So those complexities are there more. In addition to there are manufacturing complexities. We are looking at the products which are complex products, difficult to manufacture product, difficult to develop the products. Also now newer large moleculars like biosimilars, also cell and gene therapy, vaccines. So there is a diversity, totally diversity in manufacturing and complexity. So in addition to these challenges, there are other challenges we should think about is adoption of new technologies. We are talking about digital technologies. Somebody has referred just now about digital technologies to address these complexities. Also developing a future-proof talent model in pharma operations. So I have two questions related to this. And I think we should ask this question to Rajiv, who is sitting idle for a long time. We should ask him some questions so that he comes onto the... So first is the how are your respective organization addressing these challenges? Are there any learnings for the broader group here today? Second question is, are there any other structural challenges other than what we said that you believe are important for this group to recognize as we talk about the future of GMP and QVD in pharma, Indian pharma? Rajiv, please go ahead.
Thank you so much. I'm not ideal because I get a lot of input from all these discussions, and it's actually just giving us a lot of ideas about what I think everybody's on the same page. But you are right that there are complexities are there, but the most important is we are all excited to have adoption of technologies, and we are always looking forward to that. I mean, it's like nice to talk about it. Everybody is trying to say that what technology implementations are going on at individual companies. But most important is we have two things. One is the legacy products that everybody talked about, and can we transition from these legacy product technologies to the new technologies? And that transition is quite painful. First of all, we have the fear whether it will work or not. And secondly, how would we be able to convince the regulatory agencies about the transition from the legacy technologies to the new ones? So these are the things that we ask. And I'm sure that every company, when the first discussion happens, there's always something at the back of their mind, like how will you be able to convince the regulatory agencies when they ask questions? And that is the fear that everybody has. Secondly, there's a resistance to change, definitely because everybody is comfortable in their own zone, saying that it has been working well, but what will it bring it as an advantage for me? And as I said, the fear of failure will always be at the back of their mind. What will you do if it fails? Because I've been working on this technology on the existing one the last several years, and it's okay. Sometimes there have been failures, but I've been able to accommodate these failures also. But will it really help me to reduce my failures given my consultancy? Because everybody who brings in these technologies are trying to experiment on the companies, saying that some company in the remote corner of the world introduced it, so you should do it. But it's not like taking the technology from one place and just putting it over here. Every company has their own different processes to operate. So that's a big challenge. Secondly, the biggest concern now is cyber security. How are you going to handle this? And every company, whether it's not only the pharma industry, every industry is being saddled with those kind of issues. And are we able to handle it in the long run? What happens is that all my data gets wiped out. You talk about electronic data. If that data is not available later on, I have nothing to back up. At least in the recent times of the past, I would have paper-based documentation, which I can back it up. I can make copies. I can have some scanned copies. But what happens if my data, which has been generated with this new technology, is not able to get retrieved? And that is a fear that we have. And every time we have so many examples around the world where data has been lost and never been recovered. So these things will definitely be very important. And to top it up, I think we talk about these technologies. First of all, the IT infrastructure in our companies should be strengthened. I think that's the biggest challenge that we have. And if you don't augment those resources in the right manner, I think getting our technologies in the laboratories, in the manufacturing, will be quite difficult and difficult to implement in the current scenario. So I think these challenges are there. But overall, I would say that in spite of all these challenges, my experience has been quite good, and I'm excited about it because the youngsters who work with us are so excited to be part of this whole process of change. And as everybody's on smartphones and they're technologically quite savvy. So asking them to be part of this technology upgradation and changes is something that I feel that whenever we introduce something at one location, there's a lot of excitement at the other places saying that when are you going to bring these technologies to us? Because we are now, I would say, fed up with this kind of manual work, and we want to bring in this kind of thing because we are, the moment they go out of the factories, they're all on the smartphones and they are much tech savvy. But when it comes back to the offices and the factories, they're still in most of the in their earlier ways of working, and they want to bring in technologies. I think their excitement is very good. We should encourage that. We should nurture it and try to see how fast we can bring in. I think there's a huge scope, and I'm excited about the whole process that is going on now. Thank you.
Vipul, you would like to add something?
Yeah, definitely. This is very interesting subject and is a burning issue in our Indian pharma industries because complexity and diversity in generic pharma companies way of life due to the need for the business. We are not the innovators. We are the generic companies, and then our business is such like what Mr. Belapure mentioned about the thousands of SKUs and different markets, different geographies, and then different regulations. So these different regulatory bodies are having different requirement of GMP compliance and product quality. Harmonization is a marathon task. I don't think harmonization we will able to do ever because even though there are ICH, there are different forums, different activities. But if you see for one product, I can give you that we have different SFGs. What was 5 to 6 SFGs for one product because of the market demand and then the market requirement for the EMB. So coming back to the complexity and diversity, I agree with what Mr. Bilapure mentioned. What we have done in my company to overcome these issues. So I don't say that we have overcome 100%, but we have done so far so good. One thing we have focused 3 things on this to handle the complexity and diversity. One is the people. So what we have done in the people, mainly we are working on building a quality mindsets of the people to equip with the delivery of the quality product. So that is first thing which we have done. Another point which we have discussed during our another forum, capability building in focus area. We used to do the training. Now, I don't think a training is the perfect word in this current environment. So skill building, capability building, and skill sets of the operator and supervisor who handle this complexity is very, very critical and which we are focusing on. Another thing which we have done in this is organization changes at shop floor level. Because as you know that at the shop floor, like we sit in the cabins, we don't hardly take the rounds once in a month or once in 2 months, something like that. But your front runners are your operators, or your front runners are your supervisors. So if you don't encourage them, empower them, and you don't do the proper organization changes, I don't think that this complexity can be handled effectively. So what we have done the organization changes at software level to make the process more robust by incorporating dedicated workforce. Like if I have the MDI team, or I have the nasal team, or I have the TDS, my transdermal team. So those are the dedicated workforce with the supervisors. And then even though it is OSD, whether it is IR or whether it is pellet or whether it is MR, so there also we have tried to do that people are focused not only into the manufacturing, also into the analytical. So complex analysis also we have dedicated the people to do this analysis. So this is not done just like that. We have evaluated the proficiencies, attitude, the aptitude, many HR tests which we have done, and we have created the group and focus, and then try to build their capability for that complexity of this environment. We have done the governance mechanism changes for the operator and supervisor for the effective operation and review process. Because we believe, when I believe, and then my colleagues here, and we have discussed in past, that the review mechanism, if you are strong, that the first hand information, if it's reviewed, you can caught all these investigation deviations, process flows, everything can be caught in that. So we have done those type of governance structure change and mechanism. As I mentioned, we have done the skill enhancement for the activities required to perform complex dosage forms. And focus area are involved in with the help of external agencies in on-the-job training for the life cycle management. So we have involved the external agencies who are the OEMs. We use them for the on-the-job training to the operator. I will give you an example for the complex equipment which we have bought in. FAT, we have sent the operator rather than we send the QA people, because operator has to operate the machine, not the QA. So these are some of the changes which we have done in the people front. In the process front, what we have done, we have harmonized and simplified in major task is taken up by building and investing in the quality process across value stream. We have done for different business form, factory in factory concept. So this concept is like that. We have the main campus. We have the OSD, that is factory by itself. We have the nasal spray, is a factory by itself. We have the MDI, is a factory by itself. So we call it a factory concept, where the skill sets are not mix and match. So those are all dedicated equipment, dedicated processes, dedicated procedures, dedicated utilities, so on and so forth. For diversified market, separate facilities are built. So this is our strategic decision. So for the diversified market, like what Mr. Bilapuram went, like semi-regulated or the India market, we have built the different facility. We don't do the more complexity into our regulated plan. So that is one strategic decision, and then now we have almost all the companies are having the similar strategies about the market-wise plan. However, if the dosage form demands, like I cannot build a sub-gelatin facility for the EMB or for the regulated market. So such specialized dosage form like MDI, sub-gelatin, injectables, or anti-cancers. So where the processes and the complexity are still persist, but those are being taken care by the effective governance structures. Wherever the common facilities are there for the diversity of market enabling different SFGs due to different specification, common approach for GMP and compliance is taken. So in this case, for the GMP and compliance, we have taken the common approach. So we don't have the SOP for the South Africa, or we don't have the SOP for the US, or we don't have the SOP. So GMP and compliance, we have taken that is very clear that it is a common procedure and systems. But however, the product point of view, the like QBD's design and everything depend on the need for the market. Like I will give you the, even though there is one RLD, the BAB requirement for the one product is different in different market. So accordingly, we have, the impurity profile requirement is different in different markets. The particle size requirement is different markets. So all this complexity is built in our SFG coding systems, and these are done with the proper tools by the planning tools, the SFP tools, and another softwares. So another thing which we have done that embedding quality into product life cycle, like design to delivery in the each dosage forms. That is a concept which we have built in. To monitor this, we have implemented very effective stage gate reviews. So I give more emphasis in stage gate reviews. As I mentioned, review is very critical for us. So we have embedded the stage gate reviews from design to delivery. So each stage where it is designed for the which market, which product, which specification, which bioavailability, which clinical points. So all these are been reviewed at different time point from the design to the scale ups for the engineering batch to the final batches. So likewise, these stage gates are done. In these stage gate reviews, the suitability of equipment, availability of the scale up manpower, availability of the vendors, availability of the API. So all these are being discussed, and then all the issues are being taken care before we do the exhibit batch or the commercial scale batches. We have another drive which we have taken with the McKinsey recently, which we call as a simplification drive. So in this, McKinsey is our partner, where we have focused on the process. We have taken the simplification of the SOP and its harmonization. We have taken the effective planning and work allocation tools. We have taken the proper person at proper place with the help of software. So I give you example that if my operator is not trained to do the pellet manufacturing, my software is not allowing to use those equipment in that particular area. So proper person at proper place with the skill sets. Reduction in non-value adding activities and manpower, automation of the processes, reduction of paper by introducing various software. Like I will give you the example, we have recently implemented the clean software, CLEEN, and I suggest everybody to look into for the cleaning validation. So it's such a user-friendly software that the entire checklist, which is we have built based on the FMEA criteria, this checklist is built into the clean, and it gives the one by step with the visual aids to do the proper work. So some of the these things which we have done in the simplification drive in the processes. And another thing which we have done for this complexity, we have done the technology enhancement. Like during the design space, we have used, everybody is using, and then Ranjana and Zila has mentioned about QBD. So there are the softwares which are available. People are using different software, and we are using also the software for the design space identification for the effective execution, scalability, and robustness. So this scalability and robustness is a critical point, which we are really see that the complex, like what Mr. Bilapure mentioned, the simple dosage form and complex dosage form, the similar processing system. So scalability is a criteria which we are really looking at when we are doing all these stage gate reviews and then risk assessments. The selection of equipment for its suitability. Sorry?
I think we are a little bit short of time. We have got only now 15 minutes probably. And
okay,
so thank you very much. I think very elaborately you informed us. So I think Vishnu, can you take it next question?
So no, but Vipul Bhai, Raji, thanks so much for sharing. I think some of the things that I picked up, right? And again, this is a topic of complexity. I'm sure just this discussion doesn't do justice to, as I can imagine. What I picked up was talking points around how do we adopt technology, right? Shifting off from legacy systems, resistance to change. More importantly, how do you think about cybersecurity as one of the important imperatives for the organization, for organizations to think about going forward? On the other side, we also spoke about how do you think about the entire people, processes piece, governance piece. How do you think about simplifying operations, etc., to be able to usher in the new era of GMP, as it were, right? So thank you for that. I think a lot for us to chew on. in the interest of time, I did have two important topics to cover, but maybe instead of going deep, it'll be good to get quick reactions from the group, right? I think the first, we mentioned it in passing, but it's important to think about how do you unlock organization-wide collaboration, and when you think about quality by design. Especially, I mean, not just limited to say quality and manufacturing, but beyond, right? So starting from R&D to your MSTG, MSATs to even commercial, post-market, etc. Right? Just quick reactions, maybe. I'd start with Jila. Any thoughts on what your vision or perspectives on that are? What of that we've been able to achieve, or what your experience has been?
So thank you, and I try not to take a lot of time, but I really would like, when we talk about collaboration, I would like to give advice, personal advice, after 32 years of the industry. I advise our people, specifically in quality, be aware of that meaning of collaboration. Collaboration should not be abused for sake of doing what others tell you to do, especially for our junior people who don't have the absolute authority to speak up. I am very clear on that one. Collaboration doesn't mean do whatever everybody asks you. Now, let me tell you, and I get very passionate and excited about that word. So let me tell you how we are doing in regards to QBD concept, obviously. So collaboration starts with, number one, common shared goals. If you put in the common shared goals for R&D, for analytical, for QA, QC, production, engineering, supply chain. So set up that common goal, whatever your objective is, okay? From that point on, okay, you need to be aware that, again, one of the things that I think we are not doing well across the industry is really setting the priorities. We keep adding projects to our peoples, and that is one of the biggest issue for execution failures and not meeting your commitments on time. So set your priority based on those shares. Then really, again, giving dedicated resources. You cannot expect the same person to do 3 jobs, okay? Please have empathy in, I'm not even talking about a work-life balance, but be careful as what you add on individuals, because majority of our people in a different cultures, not only in India, they do not want to say no. So they do a very chopped action execution. 4, train your talents. Really train your talents, and with different kind of like a Gemba walk or the processes or analytical tool, whatever your tools are. Have an ongoing management review. It is very important. This type of a project have to come from a top down. Ideas goes from a bottom up, but the management buy-in and setup is from top down, because you can never get an organization to work toward the same common, goal, which is really a very extensive one if your CEO, chairman, MD is not buying into it. So your management buy-in and reviews, right, are just to give that people a perspective that this is important to us, right, in our culture. Then, of course, you have based on a goal, based on what you want to achieve. Like, for example, one of my objective is 10% of my product at each site annually to increase PPK or CPK. We color coded, right? From going from a red, going to orange, and then ultimately green. Okay. So, and this is not kind of like I, get up tomorrow morning and my PPK revert from 0.3 to 1.3, right? So monitoring. And then one of the most important things is learning from what has been done and reapply it, because we have been reapplying this learnings in a lot of quality by design to our new development processes. And we are actually monitoring and seeing that we have been able to change our old specification based on the data. And I don't know if our colleagues in regulatory colleagues in Europe and USA, FDA, are listening to us. We're really proud of what they have done in the past 18 months with COVID. We have seen whenever you provide the data, it's very simple to change your old specification, to remove some of redundancy. When they see the data, these colleagues in FDA, they listen to you. So we have seen a change of attitude. And so obviously, and then application of those new processes as it changes for our new products, which we have seen also the duration of our submission application has decreased because you have a right first time, more of a CMC section and application. So again, collaboration to me is a set of shared goals, listening to each other, looking at your data, apply it, and not because I am your boss, I told you, so you have to collaborate. Let's have a clear understanding in our culture. I teach that on a daily basis with my team.
Great, Sheila, thanks so much. Again, very important topic. And at that time, I'll reiterate what you started with, and I'll borrow what Ranjana just messaged to us all in the chat, right? It's more about collaboration and not collusion. Keeping that in mind, how do you look at data? What are the kind of systems you set up? What's the kind of governance, top-down buy-in? Very helpful, Jila, to at least set starting thoughts and learnings for everyone. And because Ranjana, you sent me that message, this one is this for you. The second important theme that I picked up from our discussion so far, maybe quick thoughts from Ranjana, you, and then Rajiv as well, is digital analytics and automation adoption, right? In the interest of time, maybe helpful to hear two things from both of you, right? One, how would you say or articulate what your vision of digital analytics adoption and quality going forward for you, for the industry is? And the second, where do you think we are at now? What are some of the milestones that you're proud of in terms of adoption of digital analytics? Would be helpful to just for the group to hear from you. Ranjana, maybe you can get us started.
Sure. So thanks, Vishnu. So this has also become a buzzword now. Everybody's buzzing with this digital, everything's digital. Sometimes I think people don't understand what is digital. But the way we understand it, I mean, digital, why do I need to go digital? Why do I need to automate? How is it going to make my life simpler? I think keeping that end goal in mind is, can I test faster? Can I mistake proof it? Is my compliance going to go up? These are the three objectives I have. And COVID has taught us, God forbid, if you don't have people, can I do it in absence of people or lesser, 25% staff? We all went through this. So these are some of the goals that we have in mind. There's one particular one that I want to share with the group, which we are very, very proud of. And it's now a couple of years old. And in our discussion today, as well, every discussion we talk about OOS, invalid, valid. So we came up with this tool with the help of McKinsey. And this is a tool, we call it the allocation tool. And using the allocation tool, we had to feed in a lot of data, products, what kind of products it is, what kind of robustness the product has, what are the failures. So everything about the product and what kind of chemist we have, everything about the chemist, you know, their experience, the way they come from, the geographies. So all that soft skill and the hard skills about the chemist. Then about the supervisor. What's the span of control? Do I have one supervisor and 5 chemists, or I have one supervisor and 10 chemists? Because all of these individual components gave me invalid OS, and then I don't know the reports. We all know it intuitively, but when we use this tool, it was mind-boggling, and I'm very happy to report that our success rate, I mean, we started with numbers, and now today we've gone down by 40%. And the credit goes to the tool, and it keeps getting refreshed, and we use it across Cipla sites. The tool gets refreshed because obviously people come, people go, products may change, supervisors may change, span of control. So it's a live tool, and that's huge. So taking care of my lab part. Now in my manufacturing, where do we want to go digital? We want to go digital with, can I have electronic batch records? These are again big words. When I bring in, we've already got some in some of our sites. When you have electronic batch records, what are the other problems that we didn't think of? So today's solution may be creating problems for me tomorrow, because the tool can come in very quickly. The electronic batch record can come in quickly, but have I skilled? And I think Vipul used this word, is it's no longer training, it's skilling, it's capability building. So while I have all these fancy tools, have my people been skilled to match it? Otherwise, again, I'll have new problems. Like we going into lots of tools in the laboratory, you know, electronic records, electronic paperless. We all want to go paperless. But guess what? When we are paperless and everything is recorded, none of your assignable causes which we have today, they're all gone. Wrong pipette, wrong septum, wrong solution, wrong this, wrong that. So our world is changing in front of our eyes. Another one that I'm really looking forward to, not post-facto analysis. What does this product look like? What did the CPK look like after 1 year, 2 year, 10 years? I want to know today. And these tools are already there. In my former company, we use this, and actually not 2 companies prior. So while I'm making the batch, how is my batch behaving? Can I stop the batch then and there? And that would be when we have arrived. Is then I have gained. Otherwise, I have wasted company resources, company money. I've already made the batch. What can I do with it? I can't do. I have to toss it, right? At the end of the day. And I also waste time in the lab. So the opportunities are huge, but the caveat that we all have to also take is, have we skilled our people at the same speed? Because my friends, 2 years from now, 3 years from now, we will be having sessions where the topic will be, in this new digitization world, automation world, what are the problems? And we sometimes we can't even envision those problems.
Agree,
So
absolutely agree.
Right?
Absolutely.
We have to take it with a pinch of salt. And another very important, and I know we're running out of time, is a very important mantra, which I wish it was mine, but it's not mine. It came from somebody, some young kid I was talking to who works in the automotive industry. And they have a rule, which I absolutely love. Create no defects, pass no defects, accept no defects. You know, there are 2 industries that are phenomenal. One is the automotive and one is the aviation. I mean, look at the 9 Sigma or 12 Sigma they're running at. So I think we're coming onto the cusp of a new world. Let's tread slowly. Let's go in with eyes wide open, and let's not, I mean, they sound sexy, but let's be careful. Thank you. Thanks for listening again, and thanks for the time.
Thanks, thanks, Ranjana. I think, sorry, my apologies. I think we've got a few pings from the IPA team. We are 1 minute on the clock. Rajiv, I'll overrule you. My apologies for this. I know the minute I hand over, you will also want to share your vision. And I think you also shared a lot that I'm recapping from what you've already shared with us as learnings today, right? How do you think about systems? How do you think about change that is going to get unlocked through digital analytics adoption, right? And how do you handle the resistance to change? So just on your behalf, I'm just reiterating some of the learnings on digital analytics adoption that you've already mentioned for the benefit of the group. I think given the last minute, let me just take this time to really thank our eminent and very insightful panel today. It has been an absolute pleasure hosting all of you, moderating all of you. It has been an incredible learning experience, even for me, as I'm sure it has been for all of the folks that are listening here today. And as always, as Ranjana is messaging as well now, there's never enough time to talk about such topics, right? We could talk about these for 3 days at a stretch, and I'm sure we will soon. But thank you so much for taking so much time out and having such an engaging discussion with all of us. Really appreciate it. Thank you and good evening to all.
Next time we'll have one full session for this.
Yes, absolutely.
Per topic.
Thank you. Thank you. Thank you very much.
Thank you for
Thank you.
Wow, it was such a wonderful session. It's pleasure to have such senior leaders in the discussion who not only shared on the GMP journey, but have inspired us through such beautifully crafted examples on current technology, challenges, and also being human. It's really inspiring. Thank you so much once again. Ladies and gentlemen, I would like to call upon Mr. Nilesh Gupta to deliver the closing remarks. Mr. Gupta is the Managing Director and Executive Director to the board at Lupin Limited. He also heads the Quality Forum at the IPA. Welcome, sir. Can't hear you. Sir, you aren't audible.
We are unable to hear you.
Do you hear me now?
Yes.
Okay. Perils of technology. Apologies for that. What a great session that was. And I agree with Shirish what you said. It'll be great to do a longer session on these because I think that's the real voice of quality and the voice of our operations coming through. You know, it's never enviable to be the last session on a forum such as this. But you know, in the last 2 years, the Indian pharma industry has really exemplified resilience to me in adapting to our operational needs. And collectively, Indian pharma companies have made us so proud. They took concrete steps to ensure that manufacturing plants continued running and safety and quality standards were maintained. And I see that resilience in action even today. Supply chains today are challenged with global shortages in supplies, rapid surge in input prices, and I think logistics are even more challenged than they were in pandemic times. For me, and this is what the group was talking about, one of the key takeaways from these times is that we have to unleash the potential of this industry by focusing on advancements in manufacturing technology and in quality, and in bringing in cutting edge practices to ensure reliability and compliance. I'd personally like to thank each one of the industry folks on this call today for going above and beyond during the COVID-19 pandemic and ensuring that our reputation as pharmacy for the world was held high. Patients, as you know, are at the core of our industry, and catering to their needs by ensuring continued supply of quality medicines is and will continue to be our topmost priority. In that vein, I think these workshops are spot on. As you know, the Advanced GMP Workshops are a key initiative of the IPA Quality Forum. And as you would know, the IPA Quality Forum is really a platform to exchange ideas and gain insights for industry, academia, and regulators. And increasingly, our focus on IPA Quality Forum is to really enable what we call future of work for manufacturing and for quality. This 3-day workshop was aimed at building capacity and upgrading our quality culture and quality management. I think the sessions were fantastic. They ranged from all kinds of subjects, from advancements in cold chain logistics to the well-rounded panel on global best practices and the future of GMP. And I love that our people are take to this platform and use it as an opportunity for upskilling and be up to date with technological advancements that are transforming pharma operations, especially as related to quality management. I would just like to thank everybody. I would like to thank USFDA, MHRA, WHO, and Indian authorities for their wholehearted support and participation. I'd like to specifically thank Dr. Sarah McMullen of USFDA for her video message, Dr. Somani for the keynote address, and the USFDA, MHRA, and WHO speakers. I would also like to thank PDA and USP for their continued collaboration, McKinsey, our knowledge partner, the industry experts, but most of all, you, the participants in this forum. And finally, I would like to thank the IPA Quality Forum and the staff of IPA, without which this would not be possible. Our work on taking forward quality is never going to be done, but forums such as this make sure that we stay on the right path. Wish you all the very best. Thank you.
Thank you, sir. Thank you for your inspiring words. Ladies and gentlemen, I welcome Siri sir to deliver the vote of thanks.
Thank you. I think Nilesh has made my job easy. Already he has thanked almost everybody. But let me say that this was a very interesting sessions we had actually in last 3 days. I hope all of you must have enjoyed all the sessions of 6th Advanced GMP Workshop. Now I am here to deliver the vote of thanks today on behalf of IPA. First, I would like to thank Dr. Somani, DCGI of India, for delivering keynote address and also for publishing our best practices guidelines. I thank our team of regulators across the globe, USFDA, MHRA, WHO, and CDSCO. Especially, I would like to thank Dr. Sarah McMullen of India Office of USFDA for her video message. Presentations by Dr. Rachel Harrington of USFDA, Ian Norton and Ian Jackson of MHRA, and Dr. Deuce Mubaginzi of WHO. Thank you so much for your valuable inputs. How can I forget the contributions by our collaborators, namely PDA and USP, for their active participation and cooperation? And also I would like to thank our knowledge partner, McKinsey, for providing speakers and their participation in the program. This program could not have been possible without the sponsorship by our key IPA members, Sun Pharma, Lupin, Dr. Reddy's, Zydus Cadila, Cipla, and Torrent. Also like to thank our IPA President, Samir Mehta, our Quality Forum Chief, Mr. Nilesh Gupta, and our Secretary General, Mr. Sudarshan Jain, for providing direction, guidance, and active participation. I thank all the speakers from the industry, academia, regulatory agencies, as well as service providers for sharing their valuable insights on quality management. This program was designed by core Quality Forum members, namely Dr. Ranjana Pathak of Cipla, Dr. Jila Breach from Sun Pharma, Dr. Rajiv Desai from Lupin, Mr. Vipul Doshi from Zydus, Mr. Dilkesh Shah from Torrent, Indrajit Bose from Dr. Reddy's, and Jaydev Rajpal and Vishnu Kumar Putti and Jyoti from McKinsey. We also got inputs from Mr. Ganadish Kamath. This India Pharma Forum cannot be accomplished without active participation, without your active participation. I must thank all the participants for actively engaging with IPA. I also take this opportunity to thank pharma industry employees to ensure the supply of medicine throughout this pandemic, and to all CEO of IPM member companies for their support. For the first time or second time probably, we have anchor from our own member company for this program. I would like to thank Ms. Sneha Shri from DRL for conducting the session very gracefully. Thank you for your time too. Wonderful setting of microsite and feel of conference in virtual world is all creation of our event management company, XPND. Thank you, XPND, and especially Mr. Kenneth. The architects of this program are my own IPA team, including Devang Saxena, Archana Zatkar, Roshan Tathed, Navneet, Lutz Siddhi, Vinay, and continuous encouragement from our Mr. Sudarshan Jain, our Secretary General. Thank you all and good night. Thank you.
Thank you, sir. We have now reached the end of the 6th Advanced GMP Workshop 2021. I hope the sessions were informative and the knowledge gained in the workshop serves to inspire advancements in manufacturing and quality of the pharmaceutical industry. I would like to remind you all to kindly spend few moments to provide your feedback for the sessions in the day 3. The feedback form is linked in the microsite. We'll also be sending each participant an email with the link to provide their feedback on the session. Your feedback enables us to deliver better outputs year on year. On behalf of IPA, I would like to thank you all immensely for taking out your time from your busy schedule to attend this forum. It is all of you who make the forum what it is today. IPA appreciates all your inputs and suggestions in making this year's event a grand success. We look forward to meeting you again next year and sharing many more accomplishments of the pharma industry. Until then, have a wonderful year, take care and stay safe. Thank you.